to produce those as well when you're ready. So, um, hello, see everyone. I guess actually I'll start the recording. So maybe before we dive into the homework review, just a little check-in with everybody. How's everybody doing, feeling about your final projects in the sense that has everybody been able to order now the materials that they need? Is anybody still need some help? Yeah. Okay. Okay. Yeah. So we've got the first big orders into both Thermo and Twist last week. But if you are still needing to order things, that's totally fine. But again, we need your orders fast. Anybody that still needs stuff. Does anybody still have any DNA synthesis or other things that they want to order for their projects? Yeah, ish. I guess my other question is, the DNA is going to arrive next week sometime, and hopefully the other thermo orders will as well. When the materials arrive, are you ready to go? Like, do you know what you're going to do? Like, do you know like what lab you're going to go to, what things you're going to start, how you're going to start executing? And I ask this in a very like specific way. It's like, it's one thing to feel like, okay, I've got this like list of stuff, and it's another thing to feel like, oh, I know exactly like what bench I'm going to go to, where I'm going to set my stuff up, how I'm going to start like, actually preparing my experiments. And once you're ready to go, you want to, I think, have that level of thoughtfulness about like, okay, well, okay, so let me ask you if you know where you're going to do your experiment. Do folks know? Where? Like building 68 and the fourth floor of the media lab and some other lab. Like, do you know where you're gonna do it yet? I'm probably building 68. Building 68. Okay, so what I would suggest you do is so basically this week we're going to be doing again measurement and imaging work, and that's actually very exciting. You're going to be spending Wednesday, Thursday, and Friday, or Wednesday and Thursday or Friday. You're going to pick a day. At Waters Corporation. So they've got a lab that's like kind of down the street in Kendall Square where they've got full on, high-end mass spectrometers that you're going to be able to use, which is going to be awesome. So this is the first time we've done this in How to Grow and had the students have access to these resources. So I'm actually really excited. It's oh well, nice. All these things launching all of a sudden. So it's going to be quite a it's going to be a really fun week at Waters. So you're going to be physically in person there. So recitation tomorrow is going to be at Waters, and then you're going to do your lab work Thursday or Friday at Waters. So if you haven't seen it yet, Susan sent out a sign-up sheet. So pick a lab slot, and then Waters is also very generous in offering lunch Thursday or Friday, which I would recommend. I'm sure it's going to be very good. So I would stop by, pop in for lunch. And this is also a thing too. If for those of you that are doing anything related to proteins for your work, and a lot of you are, you have an opportunity after you manufacture your proteins, if you're going to do it in cell-free or if you're going to do it with cells, you can work with Waters to potentially actually have them analyze your proteins for you. Okay, so you're going to learn about that this week, ways to analyze the structure, the sequence, the molecular weight of your proteins. It's not like there's extra credit, but if you do end up doing anything at Waters, that's awesome. We would love to see that. I think it's a great opportunity for you all just to get a hands-on experience with mass spec. Again, it's normally mass specs are incredibly expensive tools. They're like hundreds of thousands of dollars. Like it's normally not in a research flow of standard research to be able to have access to these tools. So I would highly encourage you to both Thursday or Friday go get lunch, meet the folks there. And then yeah, we'll talk about this a little bit more in the last the closing weeks of class, but go and do your projects there. I think if you've got an opportunity to analyze proteins there, I would super recommend you do it. It's a very, very special chance to go and play with their machines. It's all normally, again, it'd be very expensive to do so, but you're going to have an opportunity to basically do it for free in class. So that's one part. So I definitely recommend you do that. The other thing I'll say is that's important. So as I was saying to folks just a moment ago, once you start working on your final project, you're going to want to think through very clearly, okay, now I've got a bunch of boxes of reagents. I've got a bunch of, whatever. I've got my DNA. What are you going to do? How do you then actually start your experiment, right? So, for example, just to ask, Do any of you guys have any idea what your DNA is, how that order is arriving? Like, is it arriving dry as a powder? Is it in solution at a concentration that you know? Do you guys know? Out of curiosity? Anybody have any idea? Yeah, Ricky, what do you know what state what format your DNA is coming in?
My aging order is an outer stat.
Ah, okay, that's good. So you ordered an Agi. Okay, very cool. Okay, great. Um, I didn't place an Agi order.
No, not yet. We're not seeing that though.
Okay, okay,
I'm gonna do all of them at once because it's like we have to. Because the thing is with them, we have to they submit the order. They have to get approval from one of the people at the media lab. So it's just better to get all of them at once.
okay,
And then
okay,
from there, it takes the yeah.
okay. All right, all right. So okay, but that's good. You know it's an agar stat. So does anybody know how their DNA is arriving? You know if it's like in solution or dry. The reason I'm asking you this is because you're gonna have to figure that out. Once it gets here, you're gonna have a bunch of powder in a tube, and you'll be like, what do I do with this, right? So those are the types of things I would encourage you to start thinking about now, this week, before your reagents start arriving with next week. Just think through your protocol. Think through, okay, I've got all of these different things. How is this gonna happen? How's this gonna work? If you're doing this in building 68, where are you going to do it? How are you going to do it? And I'm going to defer a little bit to Steven on this. So you all did bioproduction last week, right? This week, you're doing the mass spectrometry and the protein work. Next week in lab, it's basically your final project time is basically starting up next week, right? There's no lab next week, right? No formal lab.
The only one that's optional is the biomaterials.
Okay, the biomaterials one. Okay.
Which, if people haven't submitted their STLs and would like to do so for the designs for the molds, please do so.
Oh, so we talked about this. Okay. All right, great. So yeah, so next week is an optional biomaterials biocement lab. But otherwise, it's time to get going on your final project experiments. I don't. Should be very soon. Yeah.
It's really gonna be those reagents.
Okay. Okay. Well, so in general, though, what I'm telling all of you is, so what do we know what the lab time is? Like how folks are going to sign up for lab time next week?
The goal is to have it from 9 to 5 when Vanessa and Anthony are there. So we'll have some set off hours for ourselves throughout the different days. Pretty much we'll try to shoot for every day at least an hour or so. But the idea is that you guys can come in the lab as soon as you have your protocol. Like again, you've all checked it off with us, so we're good on that. You guys can just come in and start experimenting. Just come in with a buddy for lab, and then you could probably get started.
Okay, so we don't need a TA in the lab to. Okay.
We've already told Anthony and Vanessa too. So
okay.
We'll get an email confirmation, but yeah, you guys should be able to have open access.
So yeah, so I think just start thinking it through what you're going to do when you get your reagents, right? It's always going to be, you're going to have a bunch of boxes on a table and then you're like, what do I do now, right? So you can start asking those questions of us. You can start kind of planning out or looking into what you want to do. So I'd recommend that y'all do that at the same time that you get going with your the waters homework this week. And I was just, for those that are just arriving, again, you've got a really special opportunity this week to learn how to work with these mass spec tools and also incorporate them in your projects, right? After you get a feel for them this week, you'll have an opportunity to work with waters to actually try and run some of your experiments with them. And so I'd highly encourage you to do that. I think that's going to be really cool data. You'll learn this week how to analyze mass spec data. And so, yeah, so just take advantage of that opportunity. It's a real rare one. But why don't we get going? Actually, does anybody have any questions about the final projects? Is anybody feeling, yeah, does anybody have any questions about stuff? Questions? Are y'all feeling confident about your final projects? Yeah, but well, I'm trying to understand, because if you have questions, it's good to ask them. There's no dumb questions, you know, so you should definitely feel free to ask if you've got them. So yeah, go ahead.
So building 68 is 9 to 5. We will have hours later than that if needed. For the upstairs facility, it's 20 hours. Okay. Yeah. We don't need a TA. If you already have that swipe, if you get swiped for that, you don't, but ideally we have a TA there. Okay.
Well, but in general, though, you can't be in there. The students can't be in there without a TA. In that, in 68 they can be there without a TA.
In 68 they can be without a TA.
Okay.
From 9 to 5, because the thing is, it's Anthony and Vanessa.
Okay. All right, good. Are you thinking about doing stuff in the fourth floor lab? Okay. So who wants to do stuff in the fourth floor lab? Does anybody have any ideas?
Because it's financial.
Okay, but what kind of experiments do you want to do there? Because that lab is only available for certain types of experiments.
Yeah, I'm not sure. I have to look at. So anything that requires a fume hood, you can't do up there because that one is not certified. The things that you use up there is mainly the open shots. So if it's like open shots related work, anything with the modules, you can do that. Or if it's for some like very simple gauge. Basically gonna do it. You can do that on there, yeah. I can look at it, maybe off the possibility. It's probably easier though to do Gibson. If you're gonna do the Gibson and transformation, yeah, the transformation is easier in 68. Yeah.
Okay, but in general though, you all feel like you're gonna do your who's gonna do their build experiments in building 68? Raise your hand if you think you're gonna do your work in 68. That should be almost everybody probably. Okay. Anybody have anything they want to do in the fourth floor here, like with robots or anything else? Shrika, maybe you? Okay. Okay.
Send us an email. Just type an email saying like, okay, requesting access or time for like 14, and then we'll figure it out.
So this week, figure it out. Figure out where you're going to do your stuff. If you want to do it in 68, make sure that you've got the stuff and equipment that you need in 68 to do it. If you want to do stuff on the fourth floor, this is your week to really kind of think through all that as your materials start to arrive. So, all right. Any other questions about final projects? And again, this is your time. Please be proactive and email us if there's still something you want to order, if there's still something that's not clear, ask. Please ask. Okay, so this is your time to do so.
Just have to give updates as soon as things arrive. Like we've already placed orders. The last ones we have to place are the hatching ones, and then whenever things arrive, basically as soon as next week, we should have a lot of things arriving, and then we'll.
Yeah. All right, then why don't we dive into the homework review? And again, so all of you have seen the sign up sheet now, right? So just for those of you that are arriving late, sign up for a spot to run your mass spec experiments at Waters. This will be, it's about a 10-minute walk from the Media Lab, but they're very generously offered lunch for everybody. So please stop by, get some food, free food if you like, and then again, pick one slot. So why don't we dive into the homework? Hopefully, this looks all weird, but hopefully the bioproduction lab went well for everyone. Is there anybody that would like to share? Anybody would like to present and offer some thoughts on how the lab went? Anybody? Or we can spin the wheel, as it were.
It should have worked for everyone. I saw everyone help, so.
Oh, did you all get your data back? Yeah. Okay, that's good. You want to are you guys discussing? You guys want to present? Do it. Looks like it. You got a lot of. I can feel it. I can feel the confidence over there. Actually, Stephen, do you mind bringing it? You can just take that chair and then
yeah,
I'm gonna grab this chair so you sit in it. Oh, thanks. Perfect.
This is yours.
Yeah, I don't know why this is all set up this way this time, but you guys want to come? You guys want to come up? Let's do it. Let's do it. All right. Yes. Thank you, bravery. All right. Where is your page? Sorry. Oh, there it is, Yolanda. Okay, great. Okay, Yolanda.
Hey, David, can you share the screen with the remote listeners?
Yes, thank you. All right. So where are we looking here?
Lab 11.
Lab. Why am I not able to see? Oh, up here. Oh, I see. Okay, great. All right. So cool. We got Sophia, Esther, Alex, Ricky, Vivek, Yolanda, and Joe. Oh, this is a big group.
There was a lot of manual labor involved. So
Okay.
This year the lab didn't take 8 hours, by the way. It
Okay.
took 3 for most people. So
Okay. All right. So yeah, why don't you go ahead, Yolanda? Feel free to walk us through this.
Yeah, so I have day 1 stuff in here, and then for day 2, I have some pictures. A lot of
Yeah, here, go ahead. I'll let you can drive.
So here you can see our labeling paradigm, which did not cause any confusion at all whatsoever. We labeled half of the tubes with hearts, and then the duplicates we labeled with both hearts and flowers because cherry blossoms in spring. And you can see Esther hard at work here, labeling our tubes beautifully. So we pipetted the like corresponding media into them and then either of two plasmids. And you can see the process here. We had a lot of fun using the serological pipettes. Oh, and then we left them in 2 different temperatures. So flowers were in 37 degrees and hearts and flowers were in 30 degrees.
So they're the same, they're just duplicates?
Yeah.
Okay.
And then this is the results after we left them overnight.
So wait, so let's let's go back over to here. So why did you pick these different combinations of media? Like what was your rationale behind these choices? Thoughts?
Don't say it was given to you guys before anyone says that.
Was this just every combination, or like what was your thinking behind this as a group? Want to chime in, Esther, Ricky? Thoughts? It's okay. It's just yeah. Go ahead.
It's every combination. We wanted to test, with the temperature, we wanted to test 30, 37 range. 37 is ideal for growth.
Yeah.
But then with 30 it's better for protein folding sometimes.
Okay.
So you want,
Holding expression. Yeah, that's the most important part.
so you want to like find which one is better for this case.
Okay.
And then with the growth media, I believe in one of the papers given it was the fructose should have been better at having the bacteria grow. So you want to test that.
Okay. Okay.
Let me reframe this section. What is the #1 goal for testing all of these? Like, what is the ultimate goal? Like the ultimate thing you're trying to maximize?
Usually the light.
Pigment.
Yep, exactly.
But so which ones would you expect to have the highest pigment, and why? Yeah. Okay, so so you're thinking then, so if we look conditions 3 and 4, so this, and this, you'd expect to have higher growth potentially? Yeah. Is that
I
also
do
your?
have a question?
Yeah, yeah.
The difference between the LV and the 2IT?
Good question.
Hmm.
Does anybody know? 2IT versus LB. You also look quickly now. I'm pretty sure that information should be in your lab protocol someplace. Anybody have any ideas? 2IT versus LB?
Does anyone know what the Y and T stand for?
Yeah. Yeast?
Yep.
Yeah, there you go. Great. So thoughts on the difference? Or any speculation on what might grow better on which one? Yeah, sure.
Well, I just also like from the results, 2YT performed better, right? Because I think it's more like nutrient dense compared to LP.
Mm-hmm. Okay, so all right, so this is some good speculations. So, and then just out of curiosity, so there's at least one other group, right? That
There should be 3.
3. Okay, got it. And did you all use the same experimental matrix, or were there any variations?
So this is the one that we gave in the lab.
Ah, okay. All right. So you basically tried every combination, but that's good too. All right. So go ahead. Feel free to keep going.
Right. So. We then put all of these samples into cuvettes and then measured we measured the absorption using the spectrophotometer.
Good. Great.
Here is our cuvette graveyard.
You have the spectrophotometer results anywhere?
Those are in a spreadsheet, which I do not have. Alex has in a spreadsheet.
Do you have them in your homework?
Yes.
Okay. All right. We can come back to it, but it'd be good to get a look at that data. But okay. All right. Let's keep going for now.
We then pipetted in each of the samples into their own centrifuge tubes.
Yeah.
And centrifuged them, so
Great.
separating out the pellet and
Yeah.
the supernatant.
Great.
We then removed the supernatant and then added more from the sample and
Mhm.
repeated until we had large enough pellets. And here's the picture.
Okay,
Um,
that looks great.
Then we added acetone to resuspend it, mixed it all up,
Uh-huh.
and we transferred the supernatant into cuvettes that diluted so that the acetone didn't melt the cuvettes, although they still might have melted them a bit.
Okay.
And then, oh, I should put a link in there to the drive with the pictures.
Alex, should we go to yours maybe and look and see? I mean, that's great. Looks like you got. Or does anybody, do you have the spectrophotometer data and or the actual
I have the spectrophotometer data.
uh
That's the only thing I have on it.
Is that it?
You have the spectrophotometer data from the first time, and I have it from the second.
Okay, well, let's just oh, okay.
Yeah.
Okay, that's okay. All right, so let's check this out.
Yeah, one quick caveat I'll say before for the recording, we actually couldn't really get for some of the spectrophotometers, it's quite difficult to get the files off the spectrophotometers.
Okay.
So just a heads up.
I just took pictures individually.
All right, so that's not working. What? Can't be found. That's weird. Okay.
And even though this lab is, a lot of it's about optimizing for these conditions, for your final projects, don't go straight into optimizing these, focus on just getting it to work once. And then you can go back to the media. In reality, we won't do a whole lot. In terms of like, and I have the questions again, comment was made about protein folding. This is not going to affect that. There are like other factors that will affect protein folding. So for example, the specific
Here?
like field.
And I'll just go to the icon mode. So it's kind of a mess in this drive, but we had some readings that appeared to work. Like for instance, maybe this one. Like you can see a clear spike right around the
Okay, so yeah, so what
wavelength
so what are you expecting here? So 11H, tell me what condition 11H is. Or anybody in your group. What is 11H? What are the conditions here?
So that is number 11, which is the beta carotene.
Uh-huh.
H, oh, and that has the LV plus fructose, and H, I believe, was H37. Yeah.
Okay. Okay. All right. And so what is this peak we're seeing at 460 here? What does that represent?
Let's see the beta carotene. The scattering the light.
Okay.
So if you read in page 4 of the file collection lab, there's a chart that shows which peaks that really like for file collection or data carotene. So
Was anybody able to plot this? Because ideally what you'd like to be able to do is like look at these curves next to each other or on top of each other.
Because these things were very little drops. Like usually the issue with these is like as soon as you try exporting it, sometimes just deletes the file.
Huh. Okay.
Like I think Ronan and I tried getting it for our measurements a few months ago, and it took us like 30 minutes just to get it exported off of these. So
Did Anthony and Vesta were they helpful?
they usually don't. So
They usually don't take the data out. Okay. Was anybody able to plot this data? Do we have plots with the data, or is it just pictures?
does anyone have thoughts? Because one thing you can also do is you can also just go and find the peak of this and then plot with it. Based on like for example, like make a bar graph or something.
So ideally, what would be good to do is for these conditions to be able to compare them, right? So if you've got, for example, your beta carotenes, it'd be nice if you're able to plot all of the beta carotene experiments all on the same graph. So then you can look and compare the peaks. So let's take, for example, or just based on just you're looking at your data, like which one of these, let's say for beta carotene for your group, which one gave the highest peak? Which one gave the kind of most the largest value here based on your data? Any ideas? So I mean, it's great that you got results, but I think what I want to see is your interpretation of the meaning here. So you got a bunch of data, which is cool, but what does this data mean? How can you interpret it?
Of the ones that we got, this might actually be the highest, but one thing we realized that we messed up on in the experimental procedure is
Mm.
like, if I show you like this one, some of them just didn't have a peak. And we
Okay,
think that
that's
happened
good.
because we didn't mix it enough when we were suspending with acetone. So like the pigment kind of just stayed in the pellet and didn't actually get into the
Okay,
secret.
so what was 6H?
This one is yeah, 2YT.
Oh, H
incubated
is like high and low, maybe, or
incubated at 37.
okay,
H-
37.
H stands for heart.
Oh, yeah, okay. But the heart versus the flower, was that a difference in temperature, or this is okay? Which one was the third? Which one was 30 and which one was 37?
I think heart was 37.
Okay. In the future, for something like that,
Write it on the.
yeah, just write the temperature on there if you don't.
On the tape should be like name, how to grow, the date, and then like either a number for like what condition you use later on, and then either. But really the first four.
I think 12 should have been the highest speed.
Yeah.
12?
12H.
Yeah, let's have a look at 12. So what's 12 versus 12H?
30 versus 37.
Okay, good. Okay, great.
Oh, yeah. And then 12 is the same treatment as 11, so both of those were high.
Okay, so 12H. Any idea why, like what's up with this part of the peak?
Just the normalization, so,
Oh.
just the baseline. Because like when you baseline this, that also can affect that. That's what's leading on.
So 12 versus 12H.
Please go ahead and update their like, should we just with the analysis stuff, so if someone go ahead, like, if you go manually with these speeds, that's fine, but like, just make a simple plot for that and then go. So I think that's something that we miss in write-ups and something we also expect for the final project.
Beyond that, I made a quick plot in the Google sheet.
Oh, great. Where should I
Like, you should have, you Google sheet, right? So go to like sheet stuff.
It's on my phone.
Oh,
In the future, just try to include all of them.
well, but I mean, this is great. I mean, again, you obviously got good looking results, which is good, but I think just if what would be like the money analysis for this would be taking all of this data and plotting it. So you can see for lycopene, right? You've got 8 different conditions, right? Let's say for lycopene and for beta carotene, you've got 8 different conditions. It'd be great to see a beta carotene graph that's got 8 conditions all mapped together, and then your lycopene with 8 conditions mapped on there. And then you can do a little just analysis and be like, why is it, do you think this certain peak was the way it was versus these other ones, right? So you're so close to that. So I guess, do you have any does anybody from this group have any insights on the data? Like, are there any kind of interesting things that you see here? Like, for example, do you have any insights about LB versus 2IT, right? Yeah, go ahead, Joffrey.
so, to be honest, I completely forgot what heart and flower heart means. So someone else in the group will have to clarify what that means. But generally, it seems like the sole flower, huh?
So 30 degrees.
Yeah, 30 degrees brew, like had a higher OD than 37 degrees. And I
For
think,
which one? For lycopene or for beta carotene?
for all, like.
Oh, okay, in general, okay. Okay.
And I think, can you go back to like the chart, like which numbers, the correspond to LB or 2I3?
This one?
Yeah.
Okay.
Okay, yeah. It seems like, yeah, definitely, LB had generally higher growth than 2IT.
Okay.
And it seems, fructose was beneficial to growth.
Okay, so dosing the fructose increased the growth.
Yeah.
Okay, all right, that's very good. Did you feel like you saw that consistently, or was that
I saw it, I think, fairly consistently. I think I think Doubtful compared to, I think, LB and 2IT.
Okay, okay. Okay. Any other observations from this group from your data? Anything? Yeah, Ricky, you're gonna no?
People find that surprising, like the fact that a lower temperature actually is expressing better. Like, I feel like for some, like the first thing you see that's counterintuitive, it's like, oh, you think the higher temperature might mean that the cola is more active and that would do something. But in this case, no, both of them seem to work better at 30. And that's also something that we've seen throughout like over the years, like working with these E. coli, but in general for a lot of these systems.
Okay, cool. Anything else from this group that you guys wanna share? Anything about the process that was surprising, or was it easy? Was it all straightforward? Did it feel like it worked well? Any challenges?
I think it just kind of emphasized to us the importance of the importance of being organized, especially with labeling and handling many things. I mean, I guess something else we mentioned was like, this is where lab automation would be.
Yeah, yeah,
We
absolutely.
have a lot
Absolutely.
of humans handling like 34
Yes.
different tubes.
That's
to
right.
get
That's
them.
right. Well, but I mean, again, it seems like overall, with whatever you organize your liquid handling, it seems like it actually worked fairly well overall, right? Or would you say no? Were there any things that did not go well because of the liquid handling?
That's all right overall.
You had a good assembly line process. Okay. Okay.
I think we only broke 3 tubes this year, which is a record. So for my collection, usually it's like 10 or 15. So like one per group.
The organization is important because with this lab it really illustrated how important it is to have different controls, to test every variable
Yeah.
on its own. And that's when I ended up with 34 different samples when on paper it seemed super straightforward.
Yeah. Yeah. Okay. Yeah, well, and actually, this is a very good kind of take it as a lesson for your final projects. Okay, so let's go back again to this chart, right? This is a really good just kind of set of experiments when you want to test a bunch of conditions, right, for your experiments, which you will want to do for your final project. Okay, in a very similar way, you'll want to organize some type of an experiment. You know, if you're doing a protein expression, for example, or if you're trying to get a cell to make a protein that you care about, or if you're going to try to do it cell-free, doing an experiment like this is useful, where you've got, you know, some type of a control in there, you've got a condition you're testing or a range of conditions you want to test. And so this is what I mean when you think about next week, once you start doing your final project, what are you going to do when you got a whole bunch of boxes in front of you? Like, what is the experiment that you're going to run, right? And so nominally, you've gotten some feedback on protocols from the TAs. So hopefully, if you do, does everybody have like a plot, like a set of conditions like this for your experiments that you want to test? Yeah. Okay.
Because I mean, your experimental plans are all pretty much ready to go to just place in motion, be like, oh, great, now you have everything set up. Maybe just add in controls for a few people, and I think it should be this fine.
Okay, but again, what's good about this is it's a good experiment, a good test in labeling, making sure that you've got all of your samples organized, and then again, when you set up a matrix like this, like obviously in this particular case, this was given to you in the lab, but you should really, for your own final project experiments, think through like what you might expect to see for these conditions you're testing. Right, this is the whole kind of a well-designed experiment. Even if it doesn't work, well, you'll learn something if you've designed it well, right? If you have a sense going into it, okay, I expect that you know, the protein production might be better at this temperature versus that temperature. If it doesn't go that way, you know, now you've got some data at least that you can help support and adjust your own hypothesis for what you thought was going to happen. So anyways, hopefully this was a good test in trying to work through an experimental matrix. All right. Any other questions or comments from your group? Any other thoughts? Okay. All right, then. Thank you guys so much. Thank you for sharing. That's great. All right. So let's do one of the other groups. It sounds like there's 2 other groups. Who else would like to share from one of the other groups? 2 other groups. Who else would like to go? Yeah? Y'all want to go? Okay, sure. Come on up. Come on up. All right. Yeah, please. There's 2 chairs up here. Come on up. And which one should I look at?
I think both of us I took inspiration. You got all the pictures formatted already.
But I highlighted, I think it's mine.
So sorry, I'm just image. Okay, Shrika. Cool. I like to just you putting the little plasma map here. That's good. Okay, great. All right. Feel free. Actually, you can drive if you like.
Sure. I mean, yeah, I think the protocol is the same as like team one. Like the first day was just like, sorry, making the samples. So we distributed them overnight.
Yeah.
And then part two, yeah, I mean, these are like the 30 to 37 degrees. And then this is our data table.
Okay,
This
there
will
you go.
come into, so so 7 and 24 were the most, which is interesting because it's actually not 2IT. For the first round of spectrometer data had the highest absorbance values. So it's like, or sorry, if you look at this table, the highest absorbance values were like the 2.099 here, and then the 2.021 here for the LYC, and then for beta carotene we had probably like the two YTs came out with the highest, like I guess like spectrometer values. And then if we go back to our data, I guess it kind of conflicts because I guess what was your final results? This is our final like wheel thing.
Great.
But the data itself, this is not labeled very well, but the highest peaks like on here correspond to 7 and 24 in the experimental conditions, which is LB for fructose and then LB plus fructose again. So generally, I think like I was like going through, I guess I have some conclusions at the end, and I was just like 30 degrees should generally lead to like better pigments because apparently like the protein is like less metabolically stressed or metabolically like stressed, and so it's like more stable. And that did seem to be the case, and so we saw like the red pigments for the lycopene and then like the orangish pigments for the beta carotene. And then I think generally 2IT did produce like better results as a whole because it is when I looked it up, they're just more nutrient dense, and so that also kind of checked out for the most part. But the data is kind of messy because I think some of the peaks didn't really like come out to be peaks; they were just like weird curves, and so there was like a little bit of data missing, so we couldn't really cross.
Okay. Okay.
But yeah, no jarringly like different conclusions in like the first.
Okay.
Peter.
But this is good. But I appreciate this. This analysis is good. So for all of you as well, just again for your final projects, when you run your experiments, you're going to get a bunch of data back, and we want you to interpret that data, right? If the experiment didn't work, that's okay, but have a thoughtful analysis or explanation for what did or did not work, right? Put some thinking in it. So this is actually this is quite nice, right? So you gave a little kind of thought on both, because essentially this experimental matrix was asking you to compare a few different things, right? Like what was it asking you to compare? Just to shout them out. What were some of the things that you were comparing here? Temperature, yes. Well, media, yes, exactly. And what's the third thing? You had a fructose supplementation, right? So you had a fructose spike, you had a 2IT versus LB, and then you had temperature, right? Those are the three things that you're basically comparing. So and then you had two different plasmids that you were looking at, right? So so I appreciate this analysis just to say, hey, this is best just in general what we saw compared for looking at the two different media conditions. This is what happened when you had fructose, and then this was the two temperature. So what would be even better would be like a quantitative analysis, because you actually have the data here. So if you actually were able to then provide a little bit of a quantitative comparison, that would be great. Again, I understand there's some challenges getting the data off of the machine, but if any one of you are able to plot that data and compare it, that would be sweet. That would be really, really nice. But this is really great. I like this a lot, being able to just show at least the cell concentration here. What's highlighted in green?
So the green is what the highest peak was in the images, like at
Okay.
the bottom. So it doesn't necessarily line up with the numbers.
Okay.
Just
Okay. Okay. No, but that's good. That's great.
This is like the cell density, right?
This is cell density, right?
That doesn't necessarily have to correlate with how much like.
Yeah.
Right. Okay. Do you want should we open up yours as well and have a look?
It's the same.
Yeah,
It's the same?
exactly. Yeah,
Okay.
control like copied with.
Yeah, I mean, we had to delete some of these things as well.
You
Right.
see like the colored, like 1500 microliter H2O versus 750, because the
Mhm,
machine wouldn't read it at a high concentration.
mhm.
And then there were a few where we had to do a third round to do like 1 to 4 dilution essentially.
Yeah.
Of the like YT and fructose ones.
Yeah.
But
Sorry, why did you have to do dilutions?
so like the machine would like not be able to read or like either too high of a concentration of the.
Oh,
receptors.
oh,
2.5 or 3.
oh, okay,
Yeah.
okay.
Yeah, so they're not like necessarily
Equal.
comparable in that sense because like they're different dilutions of the cell culture.
I see, I
Some of them
see,
are like 1 to 1, and some of them are 1 to 4.
I see, I see.
So it presumes
And I think all the ones that we did dilute were 2YT and fructose.
Yeah.
Like makes sense.
So it's,
Yeah.
so
Ah,
there's
ah.
more cell density there, which means like maybe there's more growth in the
Uh,
media.
if you
Yeah.
multiply back in your dilution,
Yes,
Yeah.
that would be like,
Four,
4 or
yeah.
2, depending on which one, like 2 or 4 times.
got it. I see, I see. Okay. Okay. Yeah, you might want to consider if you could also add another column here where if you multiply put the dilution factor back in, then you might get you could have a more comparable apples to apples comparison between these guys.
One other thing that could be interesting is that we test this by like letting it go for 24 hours. If you also go ahead and just like measure this like periodically, like every few hours, you'll see the growth curve of normal E. coli strain. Like if you ever looked at like the log and lag phase of E. coli, you'll also see that. And since this is 24 hours, it's actually more in like the stationary phase. So you might actually see where the cell's already been stressed. I mean, we do it for 24 hours because obviously we line up with lab time, but I presume that like if you do it for a little bit less, like 16 or so, you might even see higher pigment amounts, like more like brighter color and so forth. So
Yeah.
something to keep in mind too. It's like one of the assumptions that you might be making for those like cell density readouts.
Yeah. I think when I looked it up, I don't know how true this is because we didn't experimentally validate it, but apparently if you had stopped the 37, if you had stopped both of them earlier, then you'd probably have seen the 37 degrees perform better because it's like not as stressed.
Yeah.
Yeah.
And totally agree. It's like again, this is also part of the analysis where it's like you could have completely different results
Yeah.
for like as you're optimizing it, and then it brings the next question is when do you stop it? Like when do you take it out and then have the samples recovered versus like you want to let it longer, for example, to match cell.
Okay, but this is great. Yeah, great job. Good results. And yeah, thanks for the analysis. Awesome. Good work. Thank you guys. All right. Is there one or more group that wants to share how things went?
There's only one other group. so-
One other group? Yeah, you want to come up? Okay, great. And let's see this. Where's your page? Ah, John. Okay, great. Awesome. Okay.
Yeah, so my group was Alex Fun, no, I'm looking for Marcello Zian.
Okay, great.
But I think I'm gonna skip the lab protocols.
Yeah.
But I think one thing that is worth noticing, I'm not sure if this is the same condition with other groups.
By the way, strong labeling. Good job.
Oh.
This is actually really good. Just
Yeah,
for a lot of
we
clarity in terms
wrote
of
down
what's
all
going
the
on.
condition.
Yeah,
It was a long assembly line.
Yeah, but this is good though. So that way, you could pull out a tube and immediately know what you're looking at. Another way to do this is you can put numbers on here and then write down in your notebook or in your computer what it corresponds to, so you can decode it easily. But this is also quite nice.
Like you also need these labels because okay, like we went ahead and like we said okay, these like specific like rotators are ours, so that we're using it for how to grow. If you're just using your own experiments, you have to write your name, or else no one will know what it is in case let's say something breaks, or let's say like someone's trying to move some samples around. So just keep that in mind.
But good job with the labeling.
Oh yeah. I was gonna highlight that like during this step, wait, let me just do full screen. Sorry. Is that
Yeah.
like some of the like when we were sampling when we're trying to get the readings, those with fructose actually we got an error message in the beginning that says absorbance at the measuring wavelength is too high. So we actually have different dilution conditions because some of them did not work, and when we further diluted, and then some of those did not work, so we further further diluted.
Yeah, yeah,
So
yeah.
there is like different dilution conditions. I'm
Good.
not sure if
Good.
it impacts the final result.
Okay, but that's good. That's good that you included that. Good.
Yeah, specifically this one, like because the A set and B set is completely the same,
Mhm.
because everything we did duplicates, right?
Mm.
And even between them, there were like the
Ah,
variations.
so okay, so that's actually really great that you got insight here. So okay, very
for-
good.
yeah,
So
for
so
some
well, no, but I mean there should be a little bit of variation, right? But so
Yeah.
I guess one question is you could do a little bit of an analysis here because in general it looks like if these are duplicates basically, set A and set B.
Yeah.
So these look in general very close,
Yeah.
right? They're all within like, you know, whatever 5 or 10%, which is like this one though, right? So this one's 3 versus 2. So this is a pretty significant difference.
Yeah.
Just like quickly scanning
When
through.
you inoculated, how many microliters of bacteria did you use? Just 3? Okay.
But in general, the numbers are like pretty. There's only this one looks like off by quite a bit. Everything else is like
Yeah.
fairly reasonable, which is good. So that's actually a good, you know,
So
you could have an inference about how reproducible your
yeah.
samples were.
So
which is
if
good.
I like skip to the result,
Yeah.
I kind of that's a good picture.
Oh, nice. That's great. Love it.
I kind of eyeballed the highest peak.
Uh-huh.
I mean like I was gonna put all of the data on, but it's too much work. So I only put like the data set A because
Okay.
it's when I just look at the picture, it looks kind of identical.
Okay.
So I eyeballed
Very
the
nice. Great.
the highest peak
Great.
number.
Okay.
And then I'm not sure if this is the right thing to do, but I divided it to get the like pigment per cell. And then I was trying to like compare. But I think the highest one we got was number 8, which is also like a super diluted, the most diluted example at the same time.
But that's still a 2IT plus fructose, right? Which is kind of what
Yeah.
everybody's getting. Like
Yeah,
the 2IT and the fructose seem to produce the
But
highest.
that's a 37-degree one, so I'm not sure.
So but everybody else is seeing more the higher production at 30 degrees C? Was that the case? Yeah. Oh, that's
Yeah,
interesting.
yeah, I didn't plot it, but this was kind of the chart with everything.
If anybody wants to do this, just to plot it out of curiosity, I think that would be neat. If somebody wants to do that as a part of your, just to kind of wrap this up a little bit, that would be nice. But this is great though. I'm glad to see this that you added that you at least put the peak value there.
We also linked an example on last year that might be very helpful. I think Lenny's write-up from last year, 2024.
Okay.
Went through and like analyzed. So if you want some code and slash like ideas, you can go and take a look.
Any other insights from your things that you know worked well, did not work well? Any other observations? Or from anybody from your group actually? Comments?
Let me pick out what I wrote.
So yeah, fructose clearly improves. Yeah.
Difference, temperature made a difference. And
30C was best out of most conditions, 6 out of 8. Okay.
Yeah.
Okay.
Yeah.
Any other comments from your group? If not, good job. Great job, everyone. So, great. I mean, I'm glad that you all were able to get experimental results that worked. That's really, really cool. And again, hopefully you learned a little bit more about data analysis and how to kind of execute an experimental matrix. Why don't we take a break until 3 o'clock now, and then we'll come back for our lectures on measurement and imaging. So we'll take a little bit of break. So thanks everyone. Come back. Evan, you want to come up and we'll get you set up. How you doing, sir?
Good.
Good seeing you. Great.
Hey, Ben.
Doing good, dude. Doing good. Let's see. So, if you haven't already, were you able to log on to the Wi-Fi?
I am.
Okay, perfect. So what we're gonna have you do Oh, I see. I had my, I actually had my audio off on my Zoom setting. But actually, no, I think it's better for you to use this audio. Okay. Let me spotlight your video. Okay, so this is you here. Great. Okay, perfect. Let me use the restroom for that slide. And then we'll get ready to go. Perfect.
Yeah, that's all. Okay. So yeah, which is like, so. Oh, these are the individual. Yeah, usually you company them all into one. And that's. This one? Okay, okay, so, all right, we're under 5,000. That's good. Now you're doing Gibson, not prescription marketing. So, all right, so you'll put it fine for a quarter or something, but then this is probably a decent amount. And then on first, I'll say that. You have to have some sort of insight. Yeah, no, I'm trying to. But you design around the Gibson assembly, right? Yeah. But then you're saying you don't want to do a Gibson assembly, you just want to order it directly like formal from West. I can't do it. So we got to choose one or the other, right? Because if you're gonna do a Gibson, then they're not gonna do it for you. So if you want them to do it for you, then I think you have to give them a site, like this is the location we want to. I sent over all the answers. Right. So maybe let's just say that. And also, I don't need, like, apparently I think. I don't know, right? That I can order easily. Yeah. So I don't need to get the weird one. Okay, great. Also, the confirmation for the ad gene, if people could just double check that theirs is listed there, that'd be great.
All right, everyone. Let's get going.
Recording stopped.
All right. So let me just double check here. All right, cool. Lindsay, are you in the Zoom? All right, let me know once you're in there. All right, everyone. So super excited for class today. As I was sharing earlier at the beginning of class, we've got a really exciting set of lectures today. One from Dr. Evan Doherty and one from Lindsay, who's here in the back from Waters Corporation. Today, we're going to dive into the world of measurement and
Hi, sorry.
imaging.
It looks like you stopped the recording by mistake.
Oh,
Recording in progress.
Thank you very much for catching that. Appreciate that. So today we dive into the world of measurement and imaging. Our first lecture is going to be by Dr. Evan Doherty, who's a former student from George Church's lab, who has since gone on as an entrepreneur and now founded 2 different companies since his graduation from George's lab. Evan is one of our very special faculty who's been a part of How to Grow since our very first edition, which is pretty amazing. I think were you still a grad student when you started? You were, right?
Yeah.
Which is crazy. So it's amazing. Been watching Evan's career, taking fundamental discoveries and research from the lab and actually commercializing those technologies and turning them into companies that he's now sold one, and now he's on to a second one. So really, really amazing watching your career unfold over the years. And as you will see, Evan's going to give a real tour de force in terms of understanding a lot of really, really kind of key fundamentals of measurement and imaging. And we'll conclude with some of the very kind of cutting edge tools that he's been working on, both in his research and in his commercial pursuits. And then, as I shared earlier, we are so, so grateful to our colleagues from Water Corporation. We've got a whole crew here from Waters in the back, so grateful to you all for being here. And again, this is a very, very special week where we'll have a 30-minute lecture from Lindsay introducing some of the principles of mass spectrometry and some of the tools you'll be using in the lab. And as I shared at the very beginning of class, Wednesday and Thursday, Friday this week, we are so honored to be doing our work at Waters. So you're going to have a real experience in person to engage with some of these cutting edge tools. As I shared, it's an incredibly rare opportunity for all of you to have the opportunity to work with mass spec. I've actually never worked with mass spec myself in person, so I'm excited to go and have that experience. And so tomorrow's recitation at 5 o'clock will be at Waters, Cambridge Immerse, which is about a 10-minute walk from here. We gave you the address earlier. And then Thursday and Friday, you guys all have seen the sign-up sheet, and all of you have already been signing up, which is great. You'll pick one slot either Thursday or Friday. Waters, again, has very graciously offered lunch, which, for our very hungry undergrads and grad students, they are very, very excited about lunch. So thank you guys again for offering that. So we'll see you on Thursday and Friday in person for lunch, and then to pick one of those slots to run your experiments, which again are going to be connected to the self-free experiments that you all ran the other week with your GFPs, and we'll talk about that in a lot of detail over the course of this week. So that is basically the plan. And without further ado, I'm going to turn it over to Dr. Evan Doherty to bring us into the world of measurement and imaging. So everybody give a round of applause and thanks to Evan Doherty. Thank you.
And David, there's a natural break point if you do typically take a break.
Yeah, we can. Yeah, we totally can. Yeah, so we're gonna do at 4:30. We'll transition over to Lindsay. But up to you if you want to take a break before then as well. That's also works out great.
Okay, that sounds good. Yeah, we'll be very complimentary because I also will not cover any mass spectrometry. So
perfect.
But it's a very important measurement technique. And I think hopefully complementary to the overall curriculum, which is very focused historically on synthetic biology approaches and a little bit of sequencing. But I'll start on a little broader background, fundamental concepts. It's a relevant little historical journey for measurement and its importance to the field of biology. So most of the large portion of the class historically has been focused on specific goals of synthetic biology. And those are also typically a lot of your projects focus because MIT students tend to be builders. So you cover a lot of right technologies. This is kind of George's framework, things like DNA synthesis technologies and CRISPR. But read is also an important part of this. We use read technologies like omics, proteomics, transcriptomics, genomics to really confirm that we've achieved our goals. And engineering, especially in bioengineering, is a very imperfect science. And so in order to really achieve goals and build products, we really need to engage in a more traditional engineering process with a full loop, where we are able to design, build, and test iteratively to really perfect our ultimate goals. And the space has come along so far in the last 10 years. Like when I started, this was one of my first slides from the 2016 version, first inaugural year, where we were just starting to use CRISPR, applied genome editing technologies. And last year, the first approval of a CRISPR-based therapy, Casgevy, for the treatment of sickle cell disease. So in just that span of 8 or 9 years, we really went from here's a powerful new technology to actually curing diseases in people using gene editing tools. And another exciting, I don't know, have you had Colossal?
Colossal has not spoken yet.
Okay.
But yeah, Ben is an ally of the class for sure. So
it's crazy that when they started, I thought it was an insane idea, but it's amazing that they've already extincted versions of I think 2 animals, and are very close to some of their first engineered elephant kind of woolly mammoth hybrids. And from a more practical human impact standpoint, more along the lines of Casgevy, cellular, not just gene therapies, but actual cellular tissue and organ therapies are also being used in patients. So another Church Lab alumnus, Luhan Yang, formed Genesis, one of the companies that's genetically engineering pig organs to transplant into humans to cure disease. Vertex is delivering engineered pancreatic beta islet cells to cure diabetes, and there are over 10 approved immune cell therapies where people are using adoptive immune cell treatments, genetically engineered immune cells to cure a variety of cancers that are really previously uncurable. Part of my current company is in this cell therapy space, and so we're really seeing the transformation of these tools from kind of basic research to very applied, which is also scary because we don't always have a strong foundation of understanding. There's a lot of biology we don't understand. FDA made an announcement today on their accelerated approval pipeline for cell and gene therapy that they're now continuing to open the door to plausible mechanisms as part of approvals. Meaning that you can get these drugs approved without actually knowing the mechanism, as long as what you put forward is plausible, which I think again shows even though our power to engineer biology is very strong, our actual understanding of how biology works is still developing. And so a lot of times, you have these advanced cures that are genetic or cellular therapies, but we still don't have a strong understanding of really what we want to do to achieve those goals, like curing disease. And measurement technology is the missing piece there that helps us close the gap, iteratively improve those products, but also develop a more robust understanding of biology fundamentals so that we can accelerate that pipeline and deliver even more advanced cures, and do things like populate outer space and do all the things that we know synthetic biology can do in the future. So I think it's fun, in that context, to take a look back briefly at the kind of history of molecules and measurement biology. We now have a deep appreciation for the molecular complexity of biological systems, but it's, not that, ancient history. So, the concept of molecular biology really has only arisen in the last couple hundred years. The concept of protein came around first, late 1700s. Fourcroy first, described what we know as proteins, although Berzelius later, coined the word protein. That was in 1838. That's right around the same time oxygen was first discovered or named by Lavoisier. So you kind of see pairwise discovery of oxygen, concept of protein. Less than 100 years later, nucleic acids were first, described by Heidenheim and Miescher. But up until, even as late as the 1930s, if you look at something like proteins, scientists understood there was this concept, but their understanding of what they were and how they functioned was incredibly crude. In the 1930s, the leading theory of proteins was this colloidal, matter theory, that living, substances were this specialized colloidal substance, and they were classified using these very coarse, categories. There was no concept of the macromolecule. And so, to give you context for how late this is, the first television broadcasts were happening, and people were still describing protein in these, very, archaic ways. Nucleic acids, a similar story. It wasn't until the 1940s that the distinction between RNA and DNA was actually fully appreciated. That happened right around the same time that people were curing diseases, penicillin, and taking commercial airline flights in pressurized cabins. And even though scientists understood in the late 30s that chromatin was responsible for the inheritance of traits, they really didn't, so Painter and Bridges, for example, which is a famous, map of the fruit fly genome, where they were linking traits of fruit flies to the actual chromatin by imaging it and looking at these banding patterns. And when, segments went missing, they looked and saw what happened to the flies, and they were able to develop these very detailed genetic maps. But at the time, the leading theory was that the DNA component was just an architectural scaffold. It was this tetranucleotide structure they proposed, and they thought the protein on top of this scaffold was actually the information-bearing part. And it wasn't until 44 when Avery and McLeod and McCarthy did their famous experiment where they were able to isolate DNA purely and show that it could transform bacteria, give it new traits, that, you know, this idea that DNA was actually information bearing gains theme. And, you know, this was the same year they're doing the first test of atomic bomb. We saw Oppenheimer. 1940s on the protein side, Pauling and very famous, you know, scientists, they're getting closer to this structure function linkage. Looking at antibodies, they understood that the structure, the shape of the antibody was different, but they actually thought that all antibodies were the same chemically. And that different antibodies got their specificity for binding by this kind of Plato theory, where the protein was this amorphous thing, and then by touching an antigen or a protein, it could acquire this shape. And that all antibodies were actually the same chemically, but their structure was responsible for their, you know, function, and that was actually acquired physically. In 1948, Tiselius comes along with the invention of electrophoresis, and I think this is like one of these great understatements. Substances are more complex than was originally supposed. I think this stands right up there with, you know, Watson and Crick's. It has not escaped our notice that, you know, pairing could be responsible for, you know, the copying mechanism for genetic material. And, you know, in the 50s, because insulin was already being used medically at large scale, it was being purified from pig and, you know, cow pancreas and used to treat diabetes. Sanger was able to take this relatively pure and abundant protein and actually study it and develop kind of the fundamentals of really modern measurement, you know, technologies by sequencing insulin for the first time and showing that insulin as a peptide, the sequence of amino acids is actually directly responsible for both its structure and its function. He may have been doing this work and like taking a break and eating at McDonald's, which has just opened, which I think is kind of hilarious. And so you have this clear progression where advances in measurement are actually driving our understanding of biology. You know, through the 1600s, 1800s, you have microscopy being developed. Out of the German chemical industry in the 1800s, you know, you have aniline dyes where you can now stain things specifically and compare them in different samples. Through the development of crystallization and centrifugation, where you can now isolate proteins or you can separate things based on their density by spinning them really fast. You know, early 1900s, you started into invention of spectroscopy and electrophoresis, where now we're using electrical fields and interactions with, you know, light or other electromagnetic waves to characterize different molecules. And this really culminates, you know, in the 1950s, that biological systems, you know, are really composed of these three classes of large macromolecules, meaning they weigh, you know, thousands or hundreds of thousands of Daltons. And, you know, that there's this clear information flow from DNA to protein. And so, you know, Crick in 1956 describes the central dogma, and everyone's like, oh, this is awesome. It's really simple, but that didn't really last long. You know, right away, you have complexity continuing to gain steam. So in the 70s and 80s, people are discovering that actually the protein component of chromatin does bear a lot of information. You have people discovering histone modifications through the kind of characterization of the histone code and the epigenome. On the RNA side, you know, it's not just an RNA molecule, but you have the discovery of introns and splicing, which convert 20 or 30,000 human genes into hundreds of thousands of gene variants. You have RNA editing being discovered, microRNAs, different classes, and with the advent of next generation sequencing, scientists realized that if you sequence deeply enough, the whole genome is expressed into RNA. And so it's not just you have these genes that turn into RNA that do something, make a protein, but really that RNA is a whole omic universe that is very poorly characterized. I think, you know, this is interesting that introns and exons are as recent as Apple Computer. They were discovered the same year Apple was founded. And on the protein side, again, you know, protein phosphorylation, it was actually discovered in 1954. But, you know, over the next 30 years, you know, from the 70s through the late 90s, you have hundreds of post-translational modifications being discovered that further diversify thousands of protein species into millions of different variants. And so, you know, again, you have this clear path where, you know, mass spectrometry, which, you know, you'll do after this, very exciting. You know, innovations in labeling and detection, like the use of radioisotopes or fluorescent proteins or cyanide fluorescent dyes to label things specifically so we can image them and compare between samples, And really the key breakthrough is, as we discovered biochemistry, we started to leverage those discoveries to build more complex measurement technologies that actually use the discoveries of biology iteratively as new measurement tools. And so those are things like nuclease restriction reactions, like digests, the use of polymerase, to copy DNA through PCR or to do sequencing libraries, the use of antibodies, the immunogenic reactions that power our immune system to actually map proteins and quantify protein expression. And so, you get in this feedback where these biological discoveries are actually now helping us build very complex measurement technologies like the one I'll talk about in the second half of the class. But, to summarize this, we now understand, biological systems are really composed of these different types of molecules. And, they're very diverse. Some of them are listed here. And we not only have, really well mapped the landscape of this molecular diversity, but now we're starting to move into the next domain, which is how they're organized in space in order to drive biological functions, right? So you not only have genomes, you have genomes folded inside nucleus. You not only have gene expression, you have expression across different cell types in a tissue or even within a single cell. And so that move from just, listing things out and measuring all the types of things that exist to actually mapping them into space and mapping them onto mechanisms is really what defines modern biology. And, amidst all this complexity, there's really, amazing new tool. Last year, or the last couple of years, we've seen AI technologies, artificial learning, help us leverage these vast databases, measurements that we've taken to drive, understandings that humans alone really can't, reach. And I think AlphaFold is probably the signature achievement in this, because protein crystallography was able to build such large databases, tens of terabytes of atomic scale resolution images of how proteins, are folded and, what the sequence of those proteins are. We're able to feed that into algorithms to actually do predictive modeling and understand how protein folding works in a kind of from first principles manner, even though you don't always understand what's going on inside of these black box algorithms. And so, that advent of once you generate enough data that is finally resolved enough, you can start to really leverage, breakthroughs in artificial intelligence to reach, deep understanding and predictive understanding, which is what you need when you go and want to, cure a disease or something like this, right? And so this is where these measurement technologies really culminated. So, despite its ubiquity and importance, there's actually, interestingly, very little consensus in the philosophical space and the philosophy of science about what a measurement is, which I think is very interesting, as well as, what sorts of things are measurable or what conditions make measurement possible. It's, measurement is such a fundamental scientific technique that you kind of gloss over it, but it's really important to take a step back and think conceptually about measurement. I think this is a good consensus definition. Measurement is a human activity in which you interact with a system with the goal of representing aspects of that system in abstract terms, like classes and numbers. And one particular view or concept of measurement that I particularly enjoy is, derived from, MIT, Claude Shannon's information theory breakthrough, which conceptualize information as, variables that can have multiple states. And, the entropy of a variable, is related to the amount of information content. And so, a coin flip you can store as a binary 0 or 1. But in biological systems, if you apply this information theoretic account, the source of information is really anything that can be, with more than one realizable state. And so in biological information, what we're talking about here is the universe of the entire physical composition of biological samples and their localization throughout time, right? So the biological systems are information in this view, and they're really vast. And so we typically do measurement approaches by looking at very specific slices of those biological systems. So we pick out one or more analytes, which are the entities that we want to subject to measurement. We pick out the measurons, the quantities that we intend to measure, and we take these very limited slices of this kind of vast, unknowable biological information reality. And so measurement technologies are those information machines that convey information about the state of this system through some measurement Y that reflects some underlying variable X, which we can't directly access. And, measurement is the process of, decreasing the uncertainty of those biological systems. So as scientists, measurement theory says that the real world is this really fundamentally unknowable thing that's separated from us, from our mind and our understanding, but it has some real values. And measurement technologies are these, human technologies, these information machines we build that allow us to interact with those systems in order to reduce the uncertainty about their state by detecting certain values. And that's done by, sometimes violently, generating this construct where the information is encoded, is transmitted to the human user, is decoded, and is analyzed. And so, like all technologies, measurement technologies have these properties which are important for us to understand as scientists. So, one of these is error. A system has some true underlying value, and what we read out is some measured value. And the difference, between that is error. But, there are very sophisticated frameworks that scientists have to use to, really, further power, the process of science. So you have these concepts like trueness, which is an estimate of systematic error. You don't need to understand the true value to measure trueness. You only need a reference value. You have, this concept of precision, which is an estimate of random error. So, to truly obtain precision, you would need an infinite number of repeated measurements, which you never have. Accuracy really embraces both precision and trueness and is the total error of your measurement. And those performance characteristics have quantitative expressions. So for trueness, that's bias, which is that difference between a measured value obtained from a sample through multiple repeat measurements and the reference value. You have this quantitative aspect of standard deviation, which is again obtained from multiple measurements of the same sample. And those combine into this quantitative measurement uncertainty. And I think in my opinion, one of the keys to good science is having an important working knowledge of the role of error in measurement. You see a lot of things about the reproducibility crisis in science, and that's a problem as I went into with some of the new types of measurements we're able to acquire to power systems biology that to really do good science, you need to apply, cultivate an understanding of statistics and measurement theory that is working so that you can design your experiments well, you can have enough power to say things confidently. You can use statistics, to have some degree of confidence for things you publish. And if you, want to be a good scientist, I think it's worth having a very active, relationship with your understanding of measurement and its limitations. So that's error, but there are a couple other, interesting descriptions of, these measurement technologies. So, one is sensitivity. So what is sensitivity? It's, it's defined by a relationship between an input and an output curve. So if you think about a technology like RNA sequencing or single cell RNA sequencing, what is the sensitivity? This paper by my old undergraduate lab at Penn did a very thorough analysis of single cell RNA sequencing technologies to quantify this sensitivity profile so that if you have some number of molecules in the original cell, what's your probability that you will detect it at all? Because if you want to say this gene is expressed, well, if it's expressed at a low level, like 5 molecules per cell, you actually have, if you use the wrong, technology, maybe only a 50% chance of seeing it at all. Right? And so that can allow you to draw incorrect conclusions. Dynamic range is this total range of detection. What's the smallest quantity of something you can, accurately detect versus the largest amount? And these measurement technologies always have an upper limit too. Resolution is the kind of smallest detectable increment of change that you can detect. Linearity, it's very important to do quantitative analysis on data, is what function describes the relationship between your input and your output of your measurement technology. Is it linear or is it non-linear? And I think one of the most interesting also for me is hysteresis. What is the dependence of the accuracy of your measurement on the history of the system that you're studying? One of the first kind of real single cell RNA sequencing technologies used for fixed tissue samples uses the nucleus from the cell. So you can't get intact cells out of fixed tissue, but you can pop out the nucleus. And so people, pop the nucleus out of tissue and they use RNA sequencing to measure the RNA molecules. That measurement has a strong hysteresis because if a gene is just being turned on, you may detect it in the nucleus, but it hasn't yet gone to the cytoplasm. It hasn't yet turned into protein. If, on the other hand, a gene is turned off in the nucleus recently, you may not detect any nuclear RNA, but you still may have millions of copies of that protein in the cytoplasm or on the cell surface. And so when you think about using measurement technology, the hysteresis property of that technology is very important in terms of understanding the types of conclusions you can draw. And so with respect to imaging and microscopy in particular, these are very practical applications. So sensitivity, this is often derived from, the detector that we're using, the fluorophores, the, sensor, the background, the experimental technique. And so, if you do these experiments where you actually quantify fluorescent sensitivity, this is concentration of a molecule versus its signal. This is kind of raw data here. You can see that below a certain level, we basically have no confidence about the quantitative aspect of this measurement. Similarly for resolution, the actual configuration of your imaging technology, the power of the objective that you're using determines the resolution. Because when it comes to microscopy, if you're doing diffraction limited imaging, there's this formula that says the wavelength of light you're detecting divided by 2 over the times the numerical aperture of the objective, which is basically your objective's light gathering power, determines the resolution. So if you use a low numerical aperture objective, a single fluorophore photon emitter appears much larger than if you use a higher numerical objective that is more powerful at gathering light. Then things get smaller, right? Underlying reality, these are all single molecules. These are single molecule photon emitters, but they appear with greatly different resolution depending on the actual construction of your information technology, your measurement technology. And so, against this conceptual background, I want to talk now about how we build measurement technologies for biology. And I'll walk through a couple examples. So starting with, the simple ones, some of these measurement technologies really are just, detecting some property that's intrinsic to what we're measuring, like its size, its weight, or its charge, how it vibrates, its electrical conductance. And so I don't know if you do gel electrophoresis in the lab, but this is a very simple, Tiselius invented this. And so you have a very simple information generating machine. You're using an electrical field applied to a buffered hydrogel to separate molecules based on size. And the detector can be as simple as your eye. And this reveals some limited information about the biological system. It tells you the abundance of different size DNA fragments. It doesn't tell you a lot about their sequence. Maybe if you do some restriction cutting, some specific cutting, that adds in some sequence information. That's a more complex measurement technology. You can also look at an example like nanopore sequencing. I don't know if you've covered this, but
we have not.
Nanopore sequencing is a very exciting new sequencing technology where with a small amount of sample prep, you take DNA and you apply it onto a membrane that has these small nanoscopic pores connected to electrical sensors. And as the DNA molecule passes through these nanopores, it changes the conductance based on the sequence of DNA that's occluding the nanopore. And so you can see there's a very kind of simple machine. There's a protein pore, there's a pump that's pumping the DNA through. And as it passes through, it's disrupting the current that's applied over this channel. And so here you can see this kind of the electrons, the electric field being detected, and you can use this to actually directly read out the base identities of this DNA or RNA as it passes through this nanoscopic pore. More complex systems, to get more information, we have to, this is why I say often violent, like we have to actively engage in this act of ripping information out of biological systems. And we've got a couple big classes of tools that we use. One is affinity. So we use affinity, we use weak chemical bonds like hydrophobic interactions or van der Waals interactions, like the things that drive protein-protein interaction, or hydrogen bonding that drives nucleic acid hybridization, to use fluorescently tagged antibodies and bind just by protein-protein interactions against their targets. And so we can label alpha-tubulin to see the skeleton of a cell. Or we can use fluorescent, DNA probes, which will, through hydrogen bonding, bind specifically to their complementary sequences to map different regions of the fruit fly genome. This is kind of a very sophisticated version of affinity detection. Let's see, I have to go to 45 seconds. Oh, no. So in this technology, which is called Codex, that's commercialized with this company Akoya, they apply a library of antibodies that have DNA tags on them to a sample. And then they conduct a number of imaging cycles where in the first cycle, they will add in fluorescent probes that are complementary to 3 of those different antibodies. And those bind and light up those proteins, and then we can scan and acquire an image. Then we cleave off the fluorescent labels. There's the image of the first 3. Then you cleave off the fluorescent labels, strip those away, and do a second round. You add in 3 different sets of probes. And you can do this iteratively to build up this map of more and more proteins where you can measure them all at the same time. That's all based on these kind of sophisticated affinity reactions and how they built this product. The other large class are those chemical reactions that are not weak. So we're actually forming or breaking covalent or ionic bonds between molecules. And so this trick won Sanger the Nobel Prize twice. He basically did the exact same trick to sequence DNA and to sequence proteins. And the trick was creating a chemical reaction that would specifically break chemical bonds in DNA or protein, and then detect just what was on the end piece. And if you did that many, many cycles, you could build up a map of the DNA sequence or the protein sequence. And that was really through an engineering of breaking and forming different specific bonds in DNA and in proteins. And the current embodiment of sequencing also heavily uses these reactivity mechanisms where in next generation sequencing, you shear up DNA, you amplify it with adapter primer sequences, you put these on a flow cell where this flow cell has all the PCR primers embedded onto the surface. And a local bridge PCR reaction happens where these individual templates that are adapterized on the ends and contain unknown template sequence in the middle will form these highly amplified clusters in a small spot on a flow cell on a piece of glass. And that allows us to use a highly engineered polymerase reaction where we add in a DNA polymerase and fluorescent bases where each base is a different color. And these are engineered nucleotides that will only incorporate once, and then they block further extension. So we can do an extension of one base into the template and acquire an image, then cleave off that block and do a second cycle and do this many cycles until we can de novo sequence a full template read. And because these are arrayed on a piece of glass, we can use imaging to scan the whole area and simultaneously sequence millions of different molecules, right? Very sophisticated technique, but it's all built up from these other, biological reactive mechanisms like PCR. And so this has really unlocked whole omics science because we've used these building blocks and these different reactions to now map different kinds of, chemical modifications on DNA or on proteins, or looking at transcription or looking at proximity of molecules together. And, the trick for the next-gen sequencing field really is that if you have a problem, convert it into DNA sequencing. So obviously you can sequence any DNA, but you can also sequence proteins or other things by converting them into DNA information. So you have antibody libraries, you tag them with DNA. You don't detect the proteins because we can't sequence those very well. You actually detect the DNA sequences. Or things like really combining synthetic biology, we can now do things like put in tracer tags in cells, genetically engineer cells, so that they have their own barcode. And every time the cell divides, it mutates its barcode. And we can use sequencing to actually discover the lineage of cells, which daughter cell came from which parent cell, came from which parent cell, and trace using these engineered DNA barcodes the whole history, developmental history of a single cell in an animal like a mouse or a worm. And so this really has brought us into the kind of the omic era where there's an ohm for everything. and anything you want to measure. And so this is, you know, a framework for how to think about measurement technologies, how to understand their properties, how much information is extracted, what kind of information we get. And, you know, there's a kind of survey of advanced measurement technologies like nanopore sequencing, single pore sequencing, imaging mass spec, all kinds of spatial technologies like FISH and FISSEQ that I'll talk about in the second part of the lecture. And so, you know, before we break, this is kind of the role of measurement technology, but now it'll be a little more forward-looking. What do we use measurement for? So another MIT alum, Harold Morowitz, you know, kind of everyone was a philosopher of science back in the day, you know, scientists. And, you know, he thought deeply about what information do we want to get out of biological systems in order to answer the questions that we have about life. This was 1 year before the central dogma, right? So he's asking this in a very broad way. But in the last 75 years, we've now come to a consensus that the answer to questions about biology are molecular mechanisms. So this is really the currency of science is, you know, what counts for an explanation of something? It's a molecular mechanism. Until you have a mechanism, you haven't really explained something, right? And so if you look in, you know, chemistry, medicine, Nobel Prizes, they all go to mechanisms. This is how you win a Nobel Prize, figure out a mechanism. And, you know, some mechanisms are very simple, like the mechanism by which invertase converts sucrose into glucose and fructose. That mechanism is purely captured by the structure of that protein. So if you characterize the protein, you've explained the mechanism. You can win a Nobel Prize. But the things that you guys care about, we care about today, are much more complex. How does a baby turn into a person? And, you know, for complex mechanisms, this is really the field of systems biology. And so if you're not familiar, systems biology is a new field of biology that's characterized by observing biological systems through quantitative, highly dimensional, multiplex measurements, and then integrating that measurement data with our functional observations using mathematical and computational models, things like graphs and equations. And so, you know, systems biology really is a quantum leap from molecular biology or genetics. It's the new field of science. Because, you know, before you would have a feature space that was on the same order of the number of samples. You were only looking at one or two things and you had a few samples. Now you can look at billions of things at the same time, but you still only have a few samples, right? It's a completely new regime. You have fewer variables than you have equations in old biology. Now you have many more variables than you have equations. Your data is much noisier. It's much more sensitive and, you know, has batch effects, and it's relatively more expensive. And so, you know, you think about the types of experiments drive modern biology, there are things like these class comparisons. How do we categorize things from a high level? How do we understand temporal dynamics of systems? How do we survey and predict the behavior of complex systems and use network analysis to reduce this vast data into the, you know, explanatory currency of science, which is mechanisms? And, you know, the brain is a great example. If you were a biologist in 1989, your textbook had said there were 210 types of cells. One was a neuron. In 2006, there were around 145 consensus types of neurons. But with the advent of sequencing in 2015, the Kim and Eberwein groups deeply sequenced 53 individual neurons that were all the same type. They were all the pyramidal neurons. And from the very small region of the brain. So they said, okay, these 53 neurons are all identical. But when they analyzed this deep sequencing data, each of those neurons differed from every other neuron in many, many dimensions, including having many private genes. That means that neuron expressed genes that no other neurons in the sample expressed. And so you start to realize that the number of different types of cells or neurons in the brain may actually be the number of neurons in the brain. And so, you know, these emerging multi-omics approaches and building the FISSEQ in the second part of the lecture, I'll explain how the technology I developed works and how we're using it to understand these kinds of complex phenomenon and map them back into, you know, molecular mechanisms through systems biology.
So that was amazing, Evan. One suggestion, so I think maybe if you're up for it, we could actually take the break after you finish and transition. Is that okay?
That sounds great.
Why don't we take a moment though to pause for some questions? So again, I always love this lecture from you because it's just, I love how you kind of give the full history. that picture of the people eating food on the airplane, I'm like, how did we go from that to where we are now? Like, I want that airplane experience, whatever
True.
that was.
Yeah, not everything
But
got better.
I know not everything got better for sure. But anyways, questions for Evan so far. There's a whole bunch in the chat.
Great book suggestion.
Oh, which one? The information?
Mm-hmm.
Okay, good one. Yeah. Any questions in the room for Evan so far? So one thing I think it's important to just acknowledge, just based on what Evan has shared, for all of your final projects, again, you're gonna want, what I was asking you at the very beginning of class, right? Like what is the experiment that you're gonna want to run? Another similar kind of parallel question is, what are you trying to measure, right, for your final project, right? What are the specific types of data that you're trying to collect, right? As I shared with you earlier, again, grateful to colleagues from Waters. For some of you that do want to do some more proteomics, there may be some opportunities to actually do some of those measurements with Waters with some of the mass spec tools that they've got there. But what are the ways that you're going to read out information about your system? Is it going to be from a gel? Are you going to look at protein size? Are you going to look at mass? Are you going to look at the amount of protein that's being expressed? Right. So ask yourself that question. And actually, it's a good thing. I didn't even think about this, but even in the context of your homework, this might be an extra credit question we can ask. And this goes for global participants, everybody. What are you measuring for your final project? That's actually a great question for you to all consider for your homework. And then, so there's the thing that you want to measure, and then there's the tool and the mechanism for how you're going to measure it, right? What is the corresponding technology that allows you to actually measure the thing that's important for your project, right? So we'll put that out into your plant a little seed for that right now. But I think, Susan, that might be a good question we can actually augment the homework with. Any questions in the room for Evan so far? And here's a question from Kale. Are there any new math techniques in this space that excite you? That's a good question.
Oh, geez. Yeah. I'm at it in that, but I know that the Broad and Penn, there's a number of new translational math efforts that are large and funded, and I think they are bordering on kind of how we apply advanced algorithm development, AI, and I think that's been the most exciting area of the field. The mathematics, I can't say, I don't have a specific answer for, unfortunately, but it is an exciting and rapidly developing field.
Yeah.
And there's a renewed interest in big institutes that are forming around new mathematical and computational techniques to deal with this new type of measurement data that's being acquired.
That's great. Mickey asks, who are the systems bio folks doing the most advanced AI-enabled whole cell or whole organism digital twins stunt doubles? So yes, so do we have enough data now to actually create quote unquote digital twins of like real cells or other organisms?
I can't think of a specific labs, but we're getting much closer. That's been a long-standing goal of the field, is to try to purely model, living systems. And I think it was held back by the quality and resolution of the data that we were using. Now that we have these, imaging and sequencing-based technologies, I think those are rapidly improving. And I think there are some lower animal models, like bacteria or viral models, that are actually getting pretty close in terms of their predictive nature.
That's interesting. So also to connect this back, if y'all remember the synthetic minimal cell class, where Kate was lecturing about these synthetic cells they're making. That's kind of an interesting example, I think, of a type of cell, because we're building them and we're actually constructing them from parts, where you can imagine creating kind of simplified versions of that you might even be able to have a more predictive model for, given what we're learning about here. All right, Evan, why don't, if you got the juice, let's keep going. Let's get into Physique. So this is Physique always blows my mind. So hopefully you all will get excited about this one.
Yeah. So as a whole class, imaging and has been a powerful tool in Physique, because it has a specific flavor, which is the way that we get this information out of biological systems. The method of transmission is light, and light is great because natural, molecules have natural interactions with light, and engineered proteins like GFP or fluorophores interact with light. We can detect very small things, so down to nanoscopic scales through microscopy and single molecule resolution microscopy. And most importantly, I think, as a plus for the power of light as a measurement, technology tool, is that light passes between the sample and the user over vast distances. And so this ability to transmit information remotely to the human over long distances is powerful because we don't have to, futz around with like putting little probes inside of samples and things like that. Like that's just not scalable, right? But light has kind of a historical problem, is that life is very complex. We want to measure millions of things, and we want to see how they're organized in space. Types of data that sequencing or mass spec gives us, it's more like lists of things. It can detect lots of things quantitatively at the same time, but it's nothing like Google Maps, right? So mass spec data or sequencing data, it'd be like if you only got a list of, how many Mexican restaurants and miles of road were there, not really useful for getting to dinner. And, so what we want to do is build imaging technologies that actually allow us to build a Google Maps. And historically imaging, we've only looked at a few things at a time. And so, that'd be like having Google Maps that only had stop signs, but not roads. It would be useless. So how do you measure many things with light at the same time? The traditional approach has been, you have 4 colors your microscope can see, is you use a number of cycles. So like Codex, when I showed the Codex video earlier, right? We have 4 colors and we do a number of cycles. We label 4 things every time. That scales F times N. So 3 cycles, 4 colors, you can see 12 things. Another approach is colorimetric labeling. So maybe we have a microscope where we can detect, a blue versus something that's blue and yellow versus something that has 2 blues and 1 yellow. In theory, that scales, but in practice, it only gives you 2 to the power of F minus 1. So again, for 4 fluorophores, you're looking at like 15 things at a time, which is not enough. So, the thing that we pioneered at Harvard is, in George Church's lab, is using sequencing, where we make the same molecule or object light up over time and construct a barcode where this molecule is red, the same molecule is blue, the same molecule is yellow. That gives you F to the power of n scaling, which is exponential. And so now you can actually measure lots of things. And the first thing we did was actually just do RNA sequencing. And so this is RNA-specific data. So you're looking at a single fibroblast cell. Each one of those dots is from a single RNA molecule. And those color transitions are us actually sequencing the bases of RNA. And then we can process this data, just like next generation sequencing data. We build up a sequencing read that we analyze, and then we can take that and map that back to the genome and say what each one of these molecules is. And we can do that not only looking within a cell to see which RNAs are in the nucleus or in this part of the cytoplasm, but we can now look across vast distances. So many cells or tissues. So RNA-specific technology is, again, a technology I helped invent at Harvard. So I'll just walk you through very quickly how it works. So you start with a sample, and the first thing you do is you kill it. So you fix the sample chemically so nothing moves around. You permeabilize it, meaning you poke tons of holes in it, and that locks everything in place. So you have an RNA molecule that's fixed. We don't ever really detect RNA directly. We always copy it into DNA. It's more stable, and we have better kind of tools to detect DNA. So the first, step we do after that is to reverse transcribe the RNA into cDNA. So we add in primers that extend and make a copy of the RNA sequence into cDNA. When we do that, we add in a non-natural base. So this is amino allyl DUTP. It base pairs like U, but it has this primary amine hanging off of it. We can use that as a handle so that we can then cross-link that newly synthesized DNA molecule onto the protein matrix of the sample using this flexible crosslinker called BS-PEG. So this reacts with the amino allyl DUTP, and the other end cross-links with a nearby protein, holds that in place. We then liberate the cDNA from its RNA. So we free it up, we digest all the RNA out of the system. And that allows the 2 ends of the newly formed cDNA molecule to reach around and circularize. And once this forms a cDNA circle, we can then use a specialized DNA polymerase called Phi29 that will go around this circular cDNA template and generate hundreds, thousands of copies directly there in place of the original RNA. And that's called a rolling circle amplicon, or ROLINE. And again, comparing to next generation sequencing, where you have these highly amplified sequence clusters on a piece of glass that you can then sequence and detect, we generate the exact same thing. Each one of these dots is a little DNA nanoball that contains thousands of copies of that original RNA sequence, but it's in the original place inside the cell. And then we can go through and use traditional next generation sequencing chemistry, that reversible terminator chemistry, where we sequence one base at a time and take pictures. We applied that technique first to human skin cells, and this work was published in Science. And the first thing we did is said, we're just going to look at all RNA. And so we sequenced all the RNA and we analyzed it, we map it back to the genome, and what do we see? We see thousands of genes over all the major classes, right? Protein coding, non-coding, ribosomal, antisense. When we look at, bin those into functions by mapping them onto gene ontologies. These are skin cells, so we see enrichment and all these kind of ontology terms that are associated with skin function. Well, because this is sequencing data, we can actually dive into the data even more. And we can look at all of the sequencing reads that align to a certain gene, like fibronectin, FN1. And we see that we're sequencing reads from all over the RNA molecule. We're sequencing actually a SNP. So this is a homozygous SNP that was in our donor. This is George. So George is a mutant for fibronectin at this allele. He has a G to A mutation. We correctly identified that. And we can look at the alternative splicing of this gene. So fibronectin has 3 alternatively spliced exons, and we saw 2 of the 3, but not the third one. And so this made us a little nervous at first because we're like, maybe our technology is biased, it's not sensitive, it has some underlying, weakness. But the biology of this EDB exon is actually explainable because this controls the solubility of fibronectin. So fibronectin that's expressed from your liver that goes into your blood contains this exon, and that makes it soluble. Fibronectin that's expressed from skin cells is insoluble. It forms the matrix of the skin. It does not have this exon. And when we take those fibroblasts and put them into a different growth factor media, we can actually revert that developmental change and make these cells all of a sudden start expressing soluble fibronectin. And when we FISSEQ those samples, now we can start to see the expression of this exon. And so we use this technique to look at a very simple wound healing model where you grow skin cells and then you slice away a bunch of them, creating a wound. And you want to understand, skin cells don't like being alone. They like touching other skin cells. And if they detect a wound, they will grow into it. And when we apply FISSEQ to this and we look at this wound edge where you can see these cells growing into this wound and compare the gene expression here versus these cells that are, not affected by the wound, we're seeing changes in all of these gene expression. Everything in bold was upregulated or downregulated. And everything in red is specifically driving these molecular mechanisms. So these are genes that are responsible for making the cells start to migrate through the cytoskeleton, making them start to secrete new extracellular matrix, making them, to start dividing more rapidly. And so we're able to now, through spatial sequencing, start to understand this complex phenomenon of wound healing in the explanatory currency of science. We're able to map it back onto molecular genetic mechanisms that are driving this very complex cellular behavior. And so, this is a very challenging technique to do. Each experiment takes tens of hours. So we built a robot to help do it. This was the first prototype we built at Harvard. So, what the step of it looks like, you suck juice out of the sample. That's what it's doing now. You add in the reagents to do the next phase of sequencing. So you add those in, incubate them, you thermocycle it, you warm it up, cool it down, mix it, and then you couple that with a very high resolution 4-channel microscope so that you then at each base take volumetric 3D scans of the sample. And that, becomes the data that you can use to map these gene expression patterns. And we started a company called ReadCore to commercialize this. So we launched this product in 2020. And we were pretty quickly bought by 10x Genomics for a final deal value of $408 million. And they reworked our machine a little bit, but now scientists all around the world can use Xenium to apply FISSEQ technology to understand spatial gene expression changes in whatever their sample is, cells or tissues. And so I'll just end a little video from George. I'll turn on closed captioning.
Technology is structure. I mean, even function is a product of the structure. You need to know how the parts are arranged in space, like put together. It's the insight into mechanism, is how things are arranged, typically cause and effect, pathology, genetic diversity, diversity between different species. All of this is revealed when you get to high enough resolution. What limits our progress in biomedical sciences It's the technology. Science takes a big leap forward every time there's a new technology. So what's next for spatial biology is multi-omics in situ. So DNA, RNA, protein all at once, together with the cell lineage. And C2 gives you, it's relative to what? Relative to single cell, relative to microscopy without sequencing. So it combines all the best features of each. It allows you to get to subcellular resolution. It allows you to do connectomics, where say one node is connected to another. That information is typically lost when you do a single cell analysis. Similarly, when you do microscopy, a few colors, you lose the richness of identifying all the features inside of a cell. So in particular, when you're doing C2 sequencing, you now basically have every DNA, RNA, and protein molecule can have a tag that you can access either randomly or in a targeted way. What could be the impact of doing C2 methodologies? For one thing, it will allow us to identify all the parts and start addressing pathologies, how basic cellular functions go awry in infections. And then the future is also in terms of population variations, what it is that makes you different from me, why it is that one person can handle the same disease that kills another person. So that resilience is something that we'd like to understand and harness. So that one lucky person can make the entire human race lucky, revealing the mechanism. That's why it's C2.
Yeah, so, oh, no, I'm not going to turn mine off. So, the future and just in the last minute is now we're not only able to do this, build these Google Maps of living systems where we can see everything, but now we can even take the next step further and use things like CRISPR or virus libraries to not just observe natural systems with this perfect resolution, with this highly multi-flexibility to map thousands of molecules at the same time, but also to make thousands of changes simultaneously. So we can do things like CRISPR, thousands of genes, and then use sequencing to understand how each one of those changes impacts some emergent property like gene expression. And so, things like Perturb-seq, where you add in thousands of CRISPR libraries and look to see how that affects cis and trans gene regulation, you'll now be able to do this in animals and start to, make these thousands of changes in mice or in other organisms and use spatial sequencing to understand how those drive, biologies either curative or that are causing human pathologies. And so, I'm very excited that, measurement is really the essential backbone of this, new biology, systems and synthetic biology. And yeah, thank you. And there were a lot of people that, helped evolve this work, developing FISSEQ and some of the other technologies like genome and protein FISSEQ that was done at the Wies and Harvard as well as at RECORP. So.
Okay, amazing. Evan, thanks as always. This was
I was done at 4, right?
Yeah, you finished early, which is great. We love that. So why don't we have a little bit of a discussion here? So let me quickly put this back. All right. So I always ask you this every year, Evan. So what just hopefully for all of you can just take a step back and look at FISSEQ. Like it's mind-blowing to me that there is now this spatial dimension where you actually can look in X, Y, and in Z, if you look at individual slices, to start to be able to reconstruct the actual kind of geolocation, quote unquote, of all these different elements within a cell to see where all of these kind of functions are taking place. One of the other dimensions I always ask you every year is the temporal dimension, right? So we talk about space, but there's also time, right? And what's like, so FISSEQ is a to me, it's like a revolutionary technology because now instead of just like having like a tube where everything is all mixed in, you don't know where the location of anything is. Now you do have a spatial kind of registration of these different parts. Then there's this question of, how do you get temporal resolution, right? Because the real kind of crazy thing would be if you had like a whole cell and then you could see what was happening dynamically as the cell was growing or evolving or in different kind of states. So I'm always curious, Evan, if there's news on the temporal side of how that has been evolving. Yeah.
Yeah, so the first of all, the measurement is almost always an endpoint, unfortunately. And that's still largely the case. The real, meaning that you're almost always killing the sample, and you're not actually able to measure most of these technologies processes that are dynamic. That's still really the case.
Yeah.
What's exciting is, again, going back to the kind of trick of DNA sequencing, which is if you want to study something, we have amazing tools to measure DNA and sequence it. So convert the problem into a problem of DNA sequencing. And people are starting to do this to study the temporal domain and the dynamics of systems by encoding the dynamics of systems in DNA. And I think Perturb-seq is kind of one example of that, where they take cells and they put in these CRISPR libraries, and then in some of those experiments, they'll take the cells and watch them over time, image them, live cell imaging, and see what they do. So there is an example where rather than like CRISPR knockouts, they put in transcription factors that are expressing, and then they watch the cells to see what kind of cells they become. So they start with stem cells, they put in a bunch of random transcription factors, and then they watch all the cells and they see which ones become neurons or which ones become and look like liver cells. And then unfortunately, they do an endpoint where they will then collect those cells, the interesting ones, and go back and sequence to find out which genes were driving those changes. And so again, the measurement happens at the end, but you start to understand this very dynamic process of cellular development and mapping it back into genetic mechanisms, which is obviously, very temporally organized, using kind of DNA as a hack. And the other great example, I think, is Jay Shinduri's lab is one of many labs doing this lineage encoding, where they have these cassettes that they can put in the cell, and every time the cell divides, it will randomly mutate. And then at the end, they can take the mouse or the worm and grind it out, do single cell sequencing and look for those cassettes, and actually through like a change log of the genome, of that tag, they can actually map back and see the entire lineage history of that cell, of that organism back to original cells. And so, I think that's been the trend and will continue to be is, how do we use the tools that we have and add in these synthetic biology tools to build new measurement approaches that can capture and encode some of that temporal information in ways that we can detect it as experimentalists.
That's really cool. Yeah, that's really neat. Yeah, and I guess, just I'm trying to kind of connect some threads back with some of the other previous lectures. So if you remember when Chris Mason, gave that amazing lecture on astrobiology, but he also gave those kind of examples of like metasub, where whole teams of people are going around subway systems all around planet Earth and then swabbing subways, and then they do kind of metagenomic sequencing of subways. That is like an annual project, right? So they're able to get these interesting kind of data maps of specific points in subways, but then they get that data every year. So they can kind of map over time. And again, I think a lot about like the nanopore that Evan showed, which is this little kind of amazing device that you can do these single cell kind of or individual DNA molecule measurements. So are there ways to take technologies like that where you can, kind of put them around, in spatially dense areas, and then have, kind of higher frequency sampling, right? So those are just things, those are good kind of future how to grow projects even to think about, is like how you would kind of get there. But I do want to say too, I think, and Sue, maybe we can officially add this to the homework. I think for next week, for all of you, for your projects, again, to think about something that you want to measure, at least one thing that's important to your final project to measure, and then talk about your measurement strategy, I think will be really critical. So please definitely do that. And let's take another just couple of questions from the chat here before we transition over to Lindsay. Actually, Lindsay, if you want, you can come up and we can get you set up as Evan. So Evan, maybe if it's okay, we'll unplug you, but keep unplug this your stuff here, and then Lindsay, you can come up, and then we'll continue with the Q&A though. So all right, so. One part in terms of just as we transition over into mass spec, as y'all are going to see in a moment, with mass spectrometry, it's another type of endpoint measurement where you basically have like a sample in a tube and you put it into a series of interesting machines, and then you get a bunch very much interesting data that comes out, very, very detailed, high resolution data. And this set of technologies we're going to focus on is going to be related to proteins.
And I'm
Please,
not,
yeah.
I don't follow the field closely, but all right, I think a lot of the breakthroughs in mass spec have been algorithmic breakthroughs, right? The data is very complex and high dimensional.
Yeah.
And now there are actually tools to get, obviously the instruments are getting better and better, but like now you can get much better results out of that data. That may be the most powerful, even more powerful than in the genomic applications, because you have these little peptide signatures you have out of mass spec data, and now people are getting like amazing data out of them.
Well, the complexity is so rich, right, that you get so many data points that it's ideal for machine learning and other algorithms. So there's absolutely huge advancements going on there, as well as instruments.
So Lindsay, actually, I need you to go and turn your video on in a Zoom. No, perfect. I'm gonna spotlight you so you can turn your video on. Yeah, there you go. Great. And Evan, don't hang here still.
Sure.
Okay, perfect. Is your video on now?
Should be. It's off.
Perfect. Great. There it is. Awesome. Okay. So, Evan, before you go, let's just have a quick look here and other questions here in the chat. And anybody questions in the room also? Y'all are very quiet today. Questions. I'd love to hear at least one question from the room. And by the way, just to say for everybody in the room, there's an art to asking questions. Okay, there's an art to asking questions, and it's important to ask questions. It's also a 30-year grade. Okay, so make sure that I would encourage you actually every single lecture to ask a question, to try to think and formulate a question. And there's very much an art to asking questions, right? So, Evan shared a lot of stuff. I actually don't know a lot of the technical detail necessarily behind every single slide that Evan shared, but you can at least frame in your mind a part that you do understand, and then a question that connects to something you do not understand, right? So, I'm gonna actually put you all on the spot to actually ask a question. So, and there's one of you just needs to ask a question right now to help save the rest of your classmates, but we do need at least one question. All right, here we go in the back. Yeah.
I have a question for Brett.
Please, great.
So I was just wondering why you ended up going into industry rather than academia. Maybe some of your personal reasons behind that.
Great question. So the question was a career question, which is Evan's path to industry as opposed to academia.
Yeah.
So yeah, go for that, Evan.
So the main driving, force was really that a lot of these advanced technologies, kind of like a lot of science, like they get published and they never get repeated. And in measurement technologies, there's a lot of exciting, like there are thousands of sequencing methods, but a lot of them are done kind of once or by one lab. And those are tools that it's really a shame because people want to use them, but they're too challenging to use, or they're inaccessible, or a lot of the method sections are completely anemic. If you ever try to reproduce a method from science method paper, it's like impossible, which is really a shame. And I think reflects like a lot of the degradation of, science as practice. And so, we have this method PhysSeq that we developed, and I think in 2016, there were literally 2 people in the world that can do it, myself and another person in the church lab. And we couldn't always even do it successfully. Each experiment took, 2 or 3 weeks of dedicated microscope time and very specialized experimental techniques. The methods section of the PhysSeq paper, we actually published a separate Nature Methods that was, like 100 pages of detailed stuff and computer code and everything. But it wasn't something people could actually use. And it seemed like it was something that would be really valuable because I kind of believe that these scientific tools really drive the breakthroughs in biology understanding and drive impact. And the only way really to do that is through commercialization. When you commercialize something, market forces, if it's valuable and there's demand, will make it accessible to the broader public. And that's exactly what happened with PhysSeq. And so, there are other technologies that were invented around the time of PhysSeq that are similar. We weren't the only people to kind of have this idea that sequencing is a good way to get a lot of information out of samples, you know, combining this combinatoric labeling over time to look at thousands of molecules. There are only 2 or 3 of those methods that were actually commercialized. And those are the ones that everyone uses and are driving the breakthrough science. So that was like my primary motivation. And then why I stay in industry is because I think I saw that impact and also the excitement when you're working in startup industry, especially. It's kind of like being a PI, the small lab. You know, you have a lot of independence and autonomy to do research and do R&D. And so I kind of still think of what I do day to day, even as CSO at Hearten, small startup company in the cell therapy space. It's very similar, I think, to what I would be doing if I had stayed in academia and been able to get a lab of my own. So I don't think if that's what's rewarding to you, I think that you can still do those things in industry is what I found.
So one I think interesting connection, right? It's like Evan took the product of his PhD, his doctoral work, and then was able to then translate that into a commercial enterprise. And you can grow that commercial enterprise into something the size of Waters, which is a very, very significant commercial enterprise. But what you all are doing in the class as it relates to your final projects, these are the literal seeds for something that could go into a, you know, ReadCore or a Waters, right? Like, you know, the ideas that you're focusing on in class. What's really great in the class format in a university setting like this is you'll get the first kind of maybe sort of working, hopefully a little bit working prototype of a thing. That can then get further expanded upon in the context of a master's thesis or in terms of a PhD, where you then take that little seed of a thing and turn it into, you know, maybe a little sapling or a little, you know, kind of more fleshed out prototype. But again, to Evan's point, a lot of what comes out, even at the doctoral level at a place, even at MIT or Harvard, is still going to be something that you can get a cool paper out of. But is it reproducible? Can another lab take it and do something similar with it? Questionable, right? So then that next step of then going into a startup and then that first stage of commercialization really gets you to that point of now having a tool or a product that can then be used by more than just 2 people in a lab. And then really transitioning, I think, into this larger topic with Waters is then how do you create instruments that are field defining and that become just core instruments that are used by everyone that's in a larger field like proteomics. So anyways, I think that's a great question that helps us get the kind of feel for even for your class, this class here, and how we get to commercialization. Last question for Evan. I think that's a neat one from Elizabeth from San Diego. She asks, as information gathering tools get more sophisticated and you can record with more data and more sensitivity, how do you differentiate between information that is biologically relevant but rare versus noise?
Yeah,
that's a great question.
That's such a fascinating question. That's one that I've thought about a lot. You know, I write about this a little in my dissertation. If you think about things from an evolutionary perspective for a second, you know, why life, it seems kind of tautological, but you know, life is evolvable because it can evolve. It structurally is able to evolve. And one of the things, the defining characteristics of evolvable systems is exploratory behavior. So doing things that aren't necessarily advantageous or meaningful or functional, that is the fodder for evolution to grab onto those things and generate new functions or new behaviors. And from that view, I think you can consider potentially all information biological. Is there such a thing as meaningless noise? Because, you know, even a gene, like I say, the whole genome is transcribed into RNA. They've kind of found most of those RNAs, people say it's like the dark matter, or they don't do anything. But maybe they don't do anything now, but they could be left over from evolution, or they could be the beginning of some new function. And the reason that those things exist is even more fundamentally because only systems that do these random processes and generate this fodder through these exploratory behaviors or non-functional behaviors, those are the only types of systems that actually can evolve. And so I really think that I'm in the perspective that there is no such thing as noise, which is not great as an answer because, you know, maybe not right on the nose of the question. but I think it's important to collect all that data. I think you see a lot of Nobel Prizes and people who have done amazing breakthroughs where they dig into something like that. That's noise. Like microRNAs are a great example. That were ignored. People didn't think they did anything. And someone said, "Well, there's got to be some function here." Right, and studied them and figured out, okay, microRNAs are a whole class of RNA regulation that happens by these little small RNAs that everyone thought were junk or ignoring or were non-functional or were not able to be detected because a measurement technology that if your first step is to pellet your RNA, small microRNAs typically don't pellet, and so you would be using an RNA analysis technique, and the first time you try to pellet the RNA to go on to the next step, you wash away all the microRNAs. Like the measurement technology was actually not suited to even capture them. And that was, unknown. So I'm in the framework of more is better. And I think more is better will be the framework of the future especially because of AI. Yeah. I think the more data we can power into advanced algorithms, the faster we'll make discoveries of things that are unknown. But it's not particularly helpful. And so in terms of like doing a particular study or project in this class, that's a very challenging question. I think one you have to think deeply about. And that's where understanding, the technologies you're using to do your science, what its properties are, and understanding statistics are very helpful because separating signal from noise is kind of, the core process of doing scientific experimentation. And so on the other hand, you have to have a thoughtful scalpel, I think.
Yeah. But that's great. I love the you don't believe in noise. That's actually that's quite an observation. And so again, I think this is a great transition though. And Evan, that was amazing. Actually, one more big round of applause and thanks to Evan. And Evan, since you're on the Zoom, there's actually more questions for you. So feel free to just feel free in your Zoom. Yeah, just hang on and answer some more questions. So very, very excited to transition over to our next speaker. Really, really thrilled to have Lindsay joining us from Waters. Actually, you can feel free to slide over a little bit over here this way. Again, this week we're so grateful to have our colleagues from Waters Corporation here. And I think, we've been working really hard and the Waters folks have done a really amazing job over the past number of weeks in preparing for this week's lab class and the lecture and recitation today. So as I mentioned earlier, we're going to have this lecture from Lindsay now. Tomorrow, y'all are going to go in person to Waters Immerse, which is over on Binney Street. It's about a 10-minute walk from where we are here at the Media Lab. And we'll do a deeper dive tomorrow into a lot of the content today. So if we don't if everything's not totally straightened out in your mind after Lindsay's lecture today, don't worry, we'll have a full, another kind of detailed, more structured dive in tomorrow. And then again, some experiments that you'll run in person on Thursday and Friday. So, Waters, again, they are the leading company in the world creating tools like mass spec that are foundational to the life sciences, synthetic biology, and a really critical measurement technology that has enabled deep exploration and understanding, not only into the world of proteins, but so many of the different omics that Evan was referencing earlier. So with that, again, grateful to all the crew back there, Ashley and the entire team. You all have been so amazing to work with. And then Lindsay, thank you so much for being here at MIT. So everybody, please give me a round of applause and welcome Lindsay. Thank you so much for being here.
Thank you. Thank you, David. I'll go ahead and start off with a little bit of a background about myself, a little bit about the company. So I work in the research group, as do all of my colleagues in the back, which means that our focus is on designing, building prototypes, and then evolving those into the next generation of commercial instruments. And in reflecting a little bit about this class, I came to realize that what you are trying to do in the scope of molecular biology, building proteins using cellular systems, follows essentially the same pattern. You build something, you construct something, and then you test it. And you learn something about that, and then you iterate on that process. And that's what we do with our hardware prototypes, with our bits of wires and things sticking out. And that's what you ultimately do in a biological setting. So for example, you might learn how to engineer a plasmid, right? Put it into a bacterium, express it. The protein doesn't express properly or doesn't make it to the outside of the cell, it doesn't secrete. Maybe you need to change that promoter sequence. But how do you know in the first place that everything went sideways?
And so really the focus of this lecture is helping you understand a new tool that you could use in the lab this week at Waters about how to kind of test for some of those things. So several weeks ago, you guys did an in vitro translation exercise in the lab. Your TAs gave you some materials and things, you put them in a tube, and lo and behold, you produced a protein. And the question there really is, what the heck is it? Now, was your TA having a bad day and trying to mess with you and pulled the wrong vial out of the fridge or freezer? So instead of trying to make GFP, you ended up with something like a Cas9? Or was your TA just grumpy and gave you the wrong thing on purpose? Maybe not. So hopefully, by the end of this, you'll gain new tools in order to figure out for yourselves whether or not the TA screwed up on you. And so there's really three pieces of information that we're going to focus on this week that mass spectrometry can provide for you. And the first, very simplistically, is the molecular weight. And so it's not that different from the gel or the Western blot that I have shown on the screen, but it provides you with a higher degree of accuracy and precision, which improves our confidence that we made the right thing. Second question is whether or not we assembled all of those amino acids that form the primary sequence of the protein into the correct order that then yields the folded structure of the protein that is correct. So did we make the beta barrel structure that is required for GFP to fluoresce, or did we make some other wonky thing that is simply clear in solution? And of course, you guys have eyes. You can see whether or not the GFP is glowing green. But most proteins that exist are not fluorescent. So if you don't have something that has been visually indicative, you must use other tools. And that's why the analytical toolbox is expanded across fluorescence and microscopy, mass spectrometry, and a number of different things. And so I talked a little bit on the previous slide about amino acid sequence. Not only do we need the composition to be correct, but we also need the ordering of the amino acids to be perfect. And you might ask yourself why that matters. And a really good example of this is in cystic fibrosis. So in cystic fibrosis, patients are typically afflicted with these very thick mucosal membranes or linings along their lungs and other secreted fluids of their body. And by and large, 80% of cases, in fact, are caused by a single point mutation in the middle of the protein, which you can kind of see here on the screen. And so this results in a failure to transport chlorine ions from the internal side of the cell into the mucosal layer, which causes it to thicken. And so we can see that at least in certain cases, a single point mutation has a huge impact on the overall structure and critical function of the protein. So if we take this in context of the GFP, we can return to the question of, will it actually fluoresce? Can we produce something that is functionally active? And it turns out that GFP is a really interesting molecule because it has its own chromophore centered in the center of the beta barrel. So you might be familiar with something like hemoglobin, which has a heme unit. It's physically connected to the protein. Pop it out, the heme goes, but it is a physical structure. Green fluorescent protein forms its chromophore from its own amino acids. So it undergoes an autocatalytic cyclization, and if all of the amino acids are not in the correct spot, that self-catalyzation will not occur. So even changing a single amino acid could change its functional properties. So you may recall from some of your earlier lectures, maybe even some of today, that the central dogma of molecular biology is the DNA starts in the nucleus, it's transcribed into the RNA, RNA moves into the cytoplasma, from which it's translated into proteins. From there, the proteins can be additionally modified with PTMs, and so you can have different phosphorylation sites, glycosylation sites, lipidation sites added to various amino acids, providing those with different functions, different signaling behaviors, different localization within the cell can be achieved by modification of these kinds. And all of these things can be addressed by simply weighing the protein, looking at the mass and figuring out where those specific modifications can occur. And so you'll recall again that protein function is defined by 4 levels of structure. So the first is the primary sequence, the ordering of the amino acids into the little pearls along the chain that you'll see many times throughout this lecture. Next, this first layer of folding, so folding into the alpha helices, the beta sheets, some of the structured and unstructured terms that link these things together. And then finally, the twisting and folding of all of those pieces into the 3-dimensional structure, which we call the tertiary structure. Now note that we also do have quaternary structure, so the formation of multi-protein complexes, but GFP is a monomer, so we'll limit it to tertiary structure today. And so because of the different needs in the analytical world, we've evolved a number of different analytical techniques. And so as you sort of traverse your eyes from left to right across the slide, we move from relative analytical simplicity to relative analytical complexity. But with that comes an increase in the amount of information we can get from those techniques. So for example, SDS gels or SDS page gels can provide you a rough molecular weight, but it's not the most accurate, maybe plus or minus 2K or so. And as we go towards the left, we can see liquid chromatography mass spectrometry not only gives us a very precise molecular weight, but also the primary structure, as well as the localization of the modifications. Finally, on the far right, we can couple different techniques with the mass spectrometer, so in this case, ion mobility, which we'll talk more about. And from that, we can start to derive conformational information about the protein and protein complex, which really empowers us to start studying it from sort of a molecular biology perspective. So in the lecture today and the recitation tomorrow, you're going to start looking at really 3 questions. And the first is the weight correct? So you will learn throughout your lives that the scale does not lie. In this case, it does not either. So we will find by weighing directly the mass of the intact protein, whether or not it is the correct composition of amino acids. And so we can do this because all of the different amino acids have different properties. Some are basic, some are acidic, some are hydrophobic. And what that gives them is individual and unique molecular weights. And so we can use that information to assemble a collection of them together and very accurately determine if the composition is correct. Next, we can use a technique called peptide mapping to determine whether or not the primary sequence is correct. And so this will allow us to go into the individual bits of the protein and ensure that all of the amino acids are in the correct order. Finally, as a bonus exercise in the lab, you'll get to determine if the protein folded correctly. So whether or not it is comprised of all of the beta strands that form into that GFP beta barrel protein.
Really quick, just to pause on this for a second.
Sure.
So, again, just it's a simple set of concepts, but I want you to, again, think about this in the context of your final projects, right? Molecular weight, structure, and folding, right? These are 3 core attributes that you're going to be able to get out of this set of mass spec tools. And in a moment too, both for your homework this week, again, think about the things you want to measure. How many of you are actually measuring something protein-based for your projects? Or have a cool protein you're interested in? Okay, so a good number of you, right? So for all of you, as Lindsay continues on with the lecture, think a little bit about the protein that you are manufacturing for your own project, and then ask yourself as well, might mass spec be a useful tool for you in your project?
So as I get going on this, it's helpful to understand a little bit about how the LC-MS systems that we're going to talk about in the lab work. So at a very high level, it's really comprised of maybe 5 different things. Start with your sample. We use a liquid chromatography system to separate things. And then we move it into the mass spectrometer, which is comprised of an ion source, mass analyzer, a detector, and an analysis system. Important point here is the sample can really be anything. It can be the intact protein that we start with. It can be a digested version of that that's really just peptides. It can be toxins from the water. It can be pesticides off of your apples at home. It can really be almost anything in the world. And so the first piece of this is to think about what we do with that sample. So again, from your IVT reactions, you took in ribosomes, amino acids, probably a plasmid in there, maybe some other molecules. It's a really heterogeneous mixture. And so in order to handle this properly, it's helpful to try to separate all of the things up front. And so we do that by separating things as a function of time using a technique called liquid chromatography. The principle here is that the mixture of components is going to pass over what we call an LC column. It's really just a tube filled with particles that have sort of a layer on the outside of them. And the different components in that sample are going to interact with that layer differentially. So as we flush liquid over this column or this tube with beads in it, the things that don't interact very strongly come off first or elute first. And so in this diagram on the right, that's represented by this sort of green little dot. As you flow for more time, then the next set of things kind of comes off, and so on and so forth, until we can almost perfectly separate everything in the sample. And so this allows us, with the mass spectrometer, to go one by one, looking at every single analyte, which provides us a higher confidence when we look at it and allows us to get better, higher quality data. And so in the lab this week, you're going to use a technique called reverse phase chromatography. This operates on a principle based on hydrophobicity. So proteins are very hydrophobic. They have a hydrophobic core. But there are other techniques that are used that you might run across in your time or for your projects. One of these is called size exclusion chromatography, in which we separate on the basis of sizes. So next we have to move these molecules from this liquid phase in the liquid chromatography system into the gas phase, which is how the mass spectrometer operates. And so to do that, we need to first evaporate all the liquid, and we also have to convert the molecules into ions. And to do that, we use a technique called electrospray ionization. So this received a Nobel Prize in 2002 by John Fenn, and it works on a relatively simple principle. So we start with the liquid emerging from the liquid chromatography system, and we apply a very high positive potential to it. That generates a series of positive charges at the tip of the capillary, so that as the liquid kind of comes out, it generates an aerosol. It's not that different from a canister of like air spray kind of stuff. As the droplets move through, they start to dry down. The charge sits on the surface. And so over time, it builds up enough of that charge that it undergoes something called a Coulombic explosion, which is really just a fancy term for it breaking into smaller droplets, which then dry down a little bit further, break down even further, until what we're left with is bare, fully evaporated, now ions that we can control using electrostatic voltages. And so we then pull those into the mass spectrometer towards the counter electrode using kind of a descending cascade of voltage. And so one key aspect of electrospray ionization is that it can generate ions that have multiple charges. So I will go in length tomorrow at the importance of charge, because it turns out we don't actually just measure molecular weight with the mass spectrometer. We measure the mass to charge ratio. So if we don't know the charge, we do not know the molecular weight. And it turns out with electrospray, we can put multiple charges onto the ions, and it does so as a function of their relative size. So an amino acid, which is relatively small, will pick up one charge, maybe two. Something like a peptide that is comprised of 10 or 20 amino acids might gather 3, 4, up to 5 charges. And then finally, a large protein or a protein complex can have tens or hundreds of charges.
Really quickly, what was something in the lab that you already used that where charge was a critical principle involved in the technology used for measurement? It's an example of something you all, yeah.
Gel electrons.
Thank you. Beautiful. Yes, exactly.
Beautiful. And it's a similar principle, right? You're using an electric field to pull things in a certain direction. And so that's what we leverage with mass spectrometry. We have ions, and now we just manipulate them around using all of these voltages.
Another note here too, again, just as Evan was talking about, a lot of the techniques that Evan shared, there's a destructive phase where you've got a sample, it could be DNA, and you have to shear it up into little pieces, and very similar type of thing happening here, right? And so part of what's mind-blowing about mass spec is the series of different technical elements that are all pulled together so that the whole thing integrated actually works, which is kind of feels like a small miracle, but it's pretty impressive. So you can imagine here, similarly, you have a very, very complex mixture. And again, the samples that you ran with your cell-free protein synthesis, where you've got cell guts plus the protein you made, there's so much stuff in that sample. So it's actually quite complex, both first that separation phase, and then how do you then interpret the data from a very complex mixture? But anyways, Lindsay will say a lot more about that in a bit to come.
All right, so now we have ions in the gas phase. What do we do with them? And so it turns out we actually just separate them here as well. So a mass analyzer is another tool to simply separate things as a function of time. But we do it very, very rapidly because everything is in the gas phase. So whereas our liquid chromatography system might separate all of our molecules out over the course of, let's say, 10 minutes, the mass analyzer is going to do it over about 100 microseconds. So very, very quick. And the way that this kind of works is based on the conservation of energy. So we start with our sample, inject it on the left-hand side. We move it through a series of pressure gradients down to a very low vacuum region, where we suddenly accelerate it very quickly by applying a very high voltage. And so you might remember from basic kind of high school level physics that if we convert potential energy into kinetic energy, we can hold on to all of that energy. And so the kinetic energy equation here that is important is 1/2 mv squared. So the velocity and the mass are related. If we do a rearrangement, which we'll talk more about tomorrow, what ends up happening is that the smaller ions move a lot faster. So if we just hold a plate at the end of a tube and just wait for things to come in, what we'll see is that the little ions will come in, create an electric signal. Then a few milliseconds or microseconds later, the bigger ones will come in. And that's all a mass spectrum is. It's just a detector recording electric charge as a function of time after we separated it, using, in this case, a time of flight mass spectrometer. And so I'm really glad that Evan talked a lot about resolution, because it's something that we care a lot about in mass spectrometry as well. But in the context of mass spectrometry, we're talking about resolution in terms of sort of the degree to which we can separate different M over Z's. So if we look at these two kind of spectra, if you will, you can see from the left and the right that they are the same thing. But the left-hand side is a low resolution spectrum in which we can very clearly see that there are two things there, but we can't necessarily see what's underneath them. With improved resolution, we can see with much greater confidence all of the different species that comprise that larger peak. And in a lot of cases, that can be really useful, depending on what we're kind of looking at. And so because of that, we have a lot of different mass spectrometers that we make and that we use and that we sell, depending on the different applications. So starting on the far left, we have something called a single quad. It's very simple, it's relatively affordable, it provides very low resolution, but it's still a great mass spectrometer. It's great for like forensic applications. If you're testing somebody's urine for cocaine, you don't actually need to be able to resolve all that much, you just need to be able to see cocaine. The next one over is a triple quad. So this is sort of the workhorse of the industry for quantitative applications. So if you're trying to find out how much of a particular drug is in a pharmaceutical product, the triple quad is usually the way to go. So it's a relatively low resolution instrument, but it's very high sensitivity, so it can detect impurities and things like that down to a very low level. The next one over is the type of instrument that we're going to be focused on in the lab today, or not today, this week. So it's called the time of flight mass spectrometer. It's high resolution, high sensitivity. And so this is usually used for characterization type applications. So looking at your protein, identifying that sequence, all of the things that you'll do in lab. And then on the farthest right is ion mobility, which is sort of a way that we tack on an additional piece of equipment onto the mass spectrometer, giving us some of those structural pieces of information about the shape and the form of the protein. So I'm going to skip over a couple of steps here. So we talked briefly how we can get mass to charge out. We'll talk at much more length tomorrow about how we can convert that into a molecular weight. But one of the other things that we can do with the mass spectrometer is we can really physically get into that sequencing piece of it. So getting down to the peptides and understanding what the ordering of the amino acids is. And so to do that, we use a technique called fragmentation. And so the overall workflow for peptide mapping, in which fragmentation is a part, works a little bit like this. We start with the protein, it can be GFP, it can be anything. We digest it using some enzyme, trypsin is the most popular, and that provides us maybe 10, 15, 20 peptides. Those peptides are independently ionized, moved into the gas phase, and then fragmented by accelerating them into a bath gas of a neutral molecule, usually nitrogen, argon, sort of thing. And so just as in the diagram, you kind of see the weak point between the amino acids being that little linker, it turns out peptides fragment right along the peptide backbone and at almost every residue. And so the consequence of that is we see all of these different fragments that differ by a single amino acid. And so we can literally go into the mass spectrum, which is here, and calculate the mass difference or the M over Z difference between each one of these fragment ions and read off the sequence of the peptide. And so we can then do that for all 10, 20, 30 peptides that were in the digest, reassemble them back together, and make sure that we've collected enough data for every protein sequence.
Another just quick question. Anybody remember from Evan's lecture a technology that he showed that also maybe had a similar type of readout process, also looking at kind of a charge voltage type of measurement? Anybody remember? You can answer in the back, yeah.
Oh, me?
Yeah.
Oh, sorry, yeah. It was the nanopore.
Yeah, exactly. Yeah, the nanopore, very similar type of thing, right? So conceptually, it's very cool. Thanks, Joffrey.
All right, so turning back to some of the concepts, the last question is really around folding or structure. So I wanted to touch just very briefly on this technique that you'll see a little bit in the lab, which is really, really powerful. I did my PhD using this technique, so it's near and dear to my heart. And it's called ion mobility. And I'm glad you brought up electrophoresis because simplistically speaking, it's not all that different than gel electrophoresis, but in the gas phase. So you're still using an electric gradient, but instead of using sort of this mesh of STS, you're just using gas molecules. So we ionize everything, bring it into the gas phase, and then sort of pulse it into this tube that's just really pressurized. And so it's sort of, you can think of it a bit like a skydiver. So when a skydiver enters kind of that compact kind of conformation, he accelerates downward, spreads out his arms, and suddenly he's pulled way back. And so the same concept is applied with ion mobility. And so this allows us to differentiate different molecules on the basis of their shape. So one thing that we can do is differentiate different sugar isomers, which is really, really difficult to do by virtually any other technique because they're linked to one another in these very complex and interesting ways. It also allows us to look at a protein's fold. So you can see on the diagram on the right here, we call this a mobilogram. So the x-axis is drift time, usually in milliseconds. The y-axis is just kind of signal intensity, how much of that electric pulse we're measuring at any given time. And so you can see this peak on the far left, kind of a Gaussian distribution, that represents the fully native folded structure of this tetrameric protein. As we go through and unfold each one of the monomers, we can see that its functional cross-section or its size kind of increases, and it's delayed as it goes through that drift tube. And so we can use this as a tool to understand how proteins fold, what their structure looks like, kind of the different forms that they occupy. All right, so to summarize some of the things that I've talked about today, mass spectrometry is a really powerful tool. We can use it to study proteins. We can use it to study other types of molecules, metabolites, drugs, pesticides, you name it. But what it gives us is molecular weight, or specifically mass to charge, from which we can derive molecular weight. If we do some other things to the sample, digest it, fragment the peptides in the gas phase, we can derive the protein sequence. Next, we have liquid chromatography. So this uses interactions between the molecules in solution and sort of this column, stationary phase, to separate things out as a function of time. We can analyze proteins and peptides by breaking them into smaller bits. And finally, we can analyze things with ion mobility to assess their structure, shape. And so what you guys will do for your homework is a series of questions focusing first on analyzing the intact mass of the GFP, and second, by confirming the sequence of it. And you will use a system that we call the BioAccord LCMS system. It's very cute. In the lab at Waters. It's our smallest QTOF that we have. So the thing here on the left is the liquid chromatography system that I talked about. And the thing on the right is the mass spectrometer. And so with those two pieces of information, you should be able to find out whether or not you created the right sequence, whether or not it has some extra little bits on it that you didn't expect, so on and so forth. And that is all I have for you today, but I'm happy to answer any questions if you have them.
Okay, awesome. Lindsay, thank you so much. That was a fantastic overview. And so again, part of what I think is going to be exciting for this week, you're going to work with GFP, which you all know and love by now. And so part of what's going to happen in this week's lab is that you're going to work with a purified GFP sample that Waters has obtained that is going to be a much more pristine sample than what you all work with in the cell-free lab. I think our goal also is potentially to try some stuff with the student samples, right? So Stephen, the cell-free experiment that was last week with the that was done with the bioproduction lab, right?
No, that was done.
Or 2 weeks ago. 2 weeks ago. Okay. So the Waters team has actually had your samples since last week. So as a part of what's going to unfold on Thursday and Friday is going to be both working with a purified sample, but then also trying some experiments with your actual samples from that cell-free experiment. Okay. So that's going to happen on Thursday and Friday. And I really just encourage you all very, very deeply, as we've been talking about, to think about your own projects and the ways in which something like mass spec might be really enabling for what you're exploring for your work in your projects coming up. So that's a bit of the framing. And why don't we take some questions? So here, I'm just going to have a look here at the online folks, but folks in person, questions about mass spec or measurement in general too. Evan's still here, so if you guys have any questions. And again, I'm going to put y'all on the spot. I'd love to get a few in-person questions here from the folks here. Yeah, go ahead.
Each of the destructive, I think you talked about each of the destructive phases, right? Is there a point of no return? Could you easily just reverse all of them? Like, do you get back?
It's a really good question.
Actually, just to say again, the question for folks online, since this is the mic here, the question was around the destructive process for mass spec. Like, is there like a point of no return? If you destroy the sample too much, can you then not get the data?
So generally speaking, it is always destructive. Once it's in the mass spec, it's kind of done. However, the big caveat there is a technique called soft landing, where they introduce the ions and then they catch them on the other side. And they tend to use this for preparative kind of analyses for the cryo-EM. So it turns out you can purify things in the gas phase into kind of different conformations or selections, and then introduce those to other analytical techniques, which is really, really powerful.
I guess a quick follow-up, is it the conversion to the gas phase that causes like the no return? Or is it like the entire?
So that's a really good question. It's not. You can actually recollect it from the gas phase. I built a prototype a few years ago that actually did exactly that. It just tends to be once you have it inside the kind of low pressure system of the mass spectrometer, it's difficult to get it out unless you devote a lot of time to kind of landing the ions down.
Good question. There's a question in the back.
Yeah, I'm wondering, as we move from like your PhD now and looking at like protein folding mass spectrometry, how have you seen like the ML protein folding technologies create a difference in your work?
Sorry, just to repeat the question. The question was in Lindsay's experience through her PhD, as she's seen the evolution of these mass spec tools and how that has influenced the ways in which proteins are measured in your experience.
Yeah, so it's really interesting because when I was doing my PhD, AlphaFold didn't exist. So there was a really great conquest towards using mass spectrometry for structural determination, which is a lot of what I did my PhD in, using different fragmentation techniques. And now I think those still complement what we see with AlphaFold. There's a lot of proteins for which structure cannot easily be derived by NMRs or X-ray crystallographies. And so for which AlphaFold does a pretty terrible job of predicting the actual structure. So IDPs are one class. So mass spec still is active, I think, in those areas. Where you kind of see them being more complementary today is really around some of the docking work. That, of course, is the next step that AlphaFold will move on to, really figuring out how the different protein complexes interact with each other and at what interface. And mass spectrometry, I think, is still playing a key role in kind of feeding information into some of those kind of mechanisms and things.
Great question. Okay, lots of great questions online too. Teddy, my guy Teddy, asking, my impression of mass specs is that they're big and heavy. Has there been any work done in miniaturized mass spec? This is an awesome question.
Yeah,
it's an awesome question because this again, it's like this is sort of parallel to my question to Evan around what's like the low tech, the nanopore that we can put everywhere, right? So
sure.
that is the ultimate, right? Where's the low cost like mass spec that's like in my pocket?
It's ongoing everywhere all the time. So the little single quad I showed you in the middle of that deck, that is our smallest mass spec. And it can be run by a battery, so you don't have to have high-end vacuum pumps. So I think they carried it up to a cute little mountain in the UK.
Oh, wow.
Where we do our mass spec development. But there's also an entire active field around sort of like backpack mass specs. There's a professor called Graham Cooks, who's been active in this field for a long time, trying to use different techniques to kind of lower the entry point, so making ionization a little bit easier. So you can actually just use a piece of paper to ionize molecules. You just kind of like drop it on there, and we call it paper spray. It's very similar to electrospray. And so there's all of these kind of innovations happening all the time to try to get there.
That's awesome.
Which, to your answer, it's not quite there yet.
No, but that's great. I actually just saw Joe join. Joe, we were just talking about decentralized like backpack mass specs. Like when do we get the nanopore of mass specs? So glad to hear there's a lot of active work on that. That's awesome. Another question from Alfonso. How accurate is ion mobility with respect to X-ray diffraction or crystallography in determining tertiary structure?
Oh, such a good question.
Great question.
So I will caveat it with, it's very difficult to determine tertiary structure with ion mobility, but you can use it to confirm it, and you can use it to understand how the protein folds and unfolds. So as a function of kind of some of those dynamic things, which is one of the powers of it. The biggest challenge there is when the molecule moves from the liquid phase into the gas phase, it does change a little bit. We're pretty good nowadays at keeping it in its solution phase conformation, but there are small changes, and so you have to kind of account for those. And honestly, modeling and machine learning and all of those things are at play for all of those.
Okay, great. Other questions in the room? Yeah, Evan.
Sorry if I missed the answer question very beginning, but most of the time mass spec, you're talking about really powerful methods like next-gen sequencing for characterizing really samples that have been purified or collected from a sample. But I know that some people have done also imaging approaches using mass spec.
Yeah. So
I'm curious if you can talk at all about what what's going on in that field.
Sorry, the question is around imaging approaches to mass spec and what's going on in that field.
So this is actually a spot that Waters has invested heavily in. So we have two different types. They're two different kind of ionization sources, if you will. We have some of them in the lab in Cambridge that you guys will be at, so you might get to see some of it kind of work. But the idea is that you use kind of like a little emitter and you spray it at the sample that collects some of the usually lipids off of the cells and ionizes, and you can kind of see very spatially what the different lipid distributions are, what the drugs are. So for example, the pharmaceutical industry is really interested in using it to look at sort of metabolic products of a drug. So if you're interested to see where in the liver a drug goes and then how it metabolizes, it's a really, really powerful tool. Usually of mice and rats, not humans, obviously.
All right, let's do one more from Frederico. Does protein mapping entail being able to find out amino acid sequence of an array of proteins? Would it be possible to use protein sequencing to derive the genetic sequence of randomly evolved proteins or antibodies designed to fight cancer?
I don't know how to answer that one. I think going from protein sequence to gene sequence is a nebulous business because of the different codons.
Yeah.
So I don't know of any cases in which that has been done.
That's interesting. Yeah. Okay. Good one. And let's see. Is the major limiter for miniaturization the pressure limitations? That's an interesting question.
It is partially pressure limitation. I mean, a big part of the size of the mass spec is really kind of moving the ions from atmospheric pressure down to the very low pressure. So we usually kind of have this like cascading ion optics series, but it's also the mass analyzer itself can take up a fair amount of space as well as like the electronics. I mean, a small mass spec can be this big, but it might have electronics boards and fans and all of those things. And so it's getting all of the stuff surrounding it to be small that I think is usually the bigger challenge.
Okay, great. Yeah. One last question, Trika. Yeah.
Let's start, talking about like the algorithmic, like innovations in the field. How much of the field is like new methodology and like that kind of stuff as opposed to, or just like question mark, or could you see more on like the algorithm?
Yeah, so I mean,
Oh, sorry, and the question was, are the new advances more algorithmic or are they hardware?
I would say it is yes. So there are constant hardware advances being made. One of the really fun things about mass spectrometry is it's just an active field of research. The mass spectrometers are constantly getting better. For example, 5 years ago, the resolution you could get off of a mass spec was, I don't know, maybe like a couple 100,000. And now there are competitor instruments that'll go to 2 million, right? It's just evolving really quickly. On top of that, you have kind of this machine learning and data mining tools coming along. So when you can collect data that is that rich, obviously you can feed it into algorithms and do really interesting things with it from a clinical perspective or just, metabolomics of kind of studying human biology. And so all of this and it's all going very quickly. Separation tools up front are developing as well. It's wild.
It's an exciting time for mass spec.
It is.
All right, so before we wrap up, this is awesome. Just going to some quick logistics. So again, you all should have gotten this in the Slack, but this is your the sign up sheet for this week's lab. Okay, so tomorrow night, 5 o'clock, you're all going to go to 301 Binney Street. Is there anything that the students need to do? Just show up and knock on the door, and then we'll be good to go. Okay, so we'll go to Binney at 5 o'clock for recitation. So again, grateful to Waters for hosting us there. And then Thursday and Friday, I see only there's free lunch, y'all. This is like a very significant deal. So just free, and I'm confident it's going to be yummy lunch. So I encourage you all to sign up for lunch if you're able to make that happen. But please, you only need to sign up for one of these 4 lab slots Thursday and Friday. And yeah, just so excited for you all to experience mass spec in person. So can we all give one more big, huge round of applause to Lindsay and then the entire Waters team in the back for making all this happen. Thank you guys all so much. And why don't we wrap up with everyone? So Lindsay, that was amazing. And yeah, so stoked for this whole week at Waters. So see you all tomorrow, 5 o'clock in person. And remember, think about for your own final projects, what are the things you want to measure? And if there's any protein elements that you want to measure, let's talk about that because there could be some cool mass spec in your futures as well. So, all right, everyone, thank you so much.